Right now, inside your body, in every cell, there's a piece of DNA that shouldn't exist. Not DNA from your parents in the way you'd expect. Not DNA from Neanderthals or Denisovans, the ancient cousins you've probably heard about by now. Something else. Something older. Something that doesn't match any known human species, living or extinct. No skeleton has ever been found for it. No skull sitting in a museum drawer. No fossil teeth. No bone fragments. Nothing you could point to and say, there. That's them. And yet scientists are increasingly convinced this lineage was real. That it walked the earth, bred with our ancestors, and left its fingerprints scattered across the genomes of millions of people alive today. Including, quite possibly, yours. Think about that for a second. We're talking about an entire branch of humanity, one that may have existed for tens of thousands of years, that left behind zero physical evidence, except for scraps of code hidden in living people's DNA. No grave, no cave painting, no tool, just genetic echoes passed down silently, generation after generation, completely invisible until someone finally learned how to read them. And here's what should really mess with your head. Scientists don't need a skeleton to prove this population existed. They don't even need a full genome from an ancient sample. Using nothing but statistical patterns buried inside the DNA of living humans, researchers can reconstruct evidence of a population that vanished so long ago we don't even have a name for it. It's a little bit like being a detective who's never seen the criminal, never found a body, but can prove someone was in the room just by the fingerprints left on the doorknob. Except here, the fingerprints are sitting inside billions of human cells, and most people carrying them have absolutely no idea. So what does this actually mean if it's true? It means the story of human evolution that you probably learned in school, the simple branching tree with modern humans on one branch, Neanderthals on another, is wrong. Or at least, dangerously incomplete. It means there were other players in this story. Ghost populations we can't see directly but whose genetic legacy is still very much alive. And it raises questions that get stranger the longer you sit with them. Who were they? Where did they live? How did they disappear without leaving a single bone behind? And why does their DNA keep showing up in some of the most genetically diverse populations on Earth today? Before we go further into this, if you're listening to this late at night or first thing in the morning, drop a comment and tell me where you are and what time it is right now. I love seeing this thing pop up from Tokyo at midnight, Chicago at sunrise, all within the same hour. It's honestly one of my favourite parts of making these. And if you want to listen without any ads interrupting the story, follow this podcast on Spotify. It's completely free and it makes a huge difference. The link's in the description if you're already following. A 5-star review helps more than you'd think. Now let's get into what not existing actually means when your DNA says otherwise. Let's slow down for a second because we just used a phrase that sounds like a contradiction. A human species that doesn't exist but shows up in DNA. How does that even work? To make sense of it, we need to get honest about the words scientists throw around like they're interchangeable when they're really not. Species, population, lineage. These words get mashed together in headlines because mysterious ancient human lineage doesn't get clicks like new human species discovered. But if you actually want to understand what's happening in your own cells, you need to know the difference. A species technically is a group that can interbreed and produce fertile offspring, isolated enough from other groups that they're on their own evolutionary path. But here's the thing about ancient humans. They didn't respect that definition even a little bit. Modern humans, Neanderthals, Denisovans, they were all interbreeding with each other for tens of thousands of years. By the strict biological definition, that means they probably weren't fully separate species at all. They were more like deeply diverged populations, cousins who'd been apart long enough to look different, act different, maybe even think a little different, but not long enough to lose the ability to have kids together who could have kids of their own. That's why scientists increasingly use the word lineage instead. A lineage is just a distinct genetic thread, a population that split off from the rest of us and evolved on its own trajectory for some stretch of time, whether that's 10,000 years or 500,000. It doesn't require a formal species label. It doesn't require a skeleton. It just requires evidence that a group existed, evolved somewhat independently, and left a genetic signature behind. And that's exactly what we're talking about when we say there's DNA in millions of people that doesn't match anything we've found in the fossil record. So here's the part that should really change how you think about this. A lineage can be completely real, completely historical, walking around, eating, breeding, dying, raising children for tens of thousands of years, and still be totally unknown in the fossil record. That's not some wild exception. That's actually the normal state of affairs for most of human evolutionary history. Fossilization is an incredibly rare event. You need the right conditions, the right burial, the right sediment, the right lack of scavengers, the right geological luck to preserve bone long enough for us to dig it up hundreds of thousands of years later. Most individuals who ever lived, across every hominin lineage, left absolutely nothing behind. No bones, no teeth, nothing but the fact that they had children, and those children had children, and eventually some fragment of their genome ended up in you. This is where scientists introduce a concept that sounds almost like something out of a ghost story, and honestly, it kind of is. They call it a ghost population. A ghost population is a group inferred purely from its genetic fingerprints in living or ancient DNA, with no fossil evidence to back it up directly. No skull, no femur, nothing you could put in a display case. Just statistical patterns, unusual chunks of DNA that don't match any known ancestor, that behave like they came from somewhere else, some other branch of the human family tree that we've never physically found. Think about how strange that actually is. Geneticists are essentially saying, we can prove someone was here even though we've never seen them, never touched their bones, never found their tools. It's a little like being able to prove a house was on fire just by testing the smoke that drifted into a neighbouring town, decades after the flames went out. The house is gone. Nobody saw it burn. But the smoke tells you unmistakably that a fire happened, roughly where and roughly how big. Now, it's important to draw a clear line here, because these three things get blurred together constantly in pop science headlines. First, there's an unknown lineage. That means we have genetic evidence of a population, but we don't know who they were, when exactly they lived, or where. Second, there's undiscovered fossils. That means the physical evidence exists somewhere, buried in a cave or a riverbed. We simply haven't excavated yet. And third, there are extinct branches we already know about, like Neanderthals and Denisovans, where we do have bones, and the genetic and fossil evidence line up nicely. The mystery lineage we're talking about in this story falls into that first category. It's not that scientists found a skeleton and are waiting to classify it. It's that they found genetic patterns that don't match anything currently known, anything with a name, anything with a fossil to its credit. It could mean there are fossils out there waiting to be found that will eventually match up with this signal. Or it could mean this population is one we may never physically recover, because maybe they lived somewhere with terrible preservation conditions, or maybe their population was too small and scattered to ever leave enough remains for us to stumble across. And here's a caution worth holding on to as we go deeper into this story, because we're going to come back to it again and again. A strange genetic signal doesn't automatically mean a brand new mystery species. Sometimes it does, but sometimes it's something more mundane, quietly waiting to be explained by mutation rates that vary more than we thought, by ancient population structure within a single lineage, by statistical artifacts in how the DNA was sequenced or analyzed. Geneticists have been burned before by exciting signals that turned out to have boring explanations. That doesn't make this particular mystery less exciting. It just means we should hold on to our excitement a little carefully, the way you'd hold a hot cup of coffee enjoying it, but ready for it to surprise you. So keep this framework in your head as we go forward. Species is a messy, almost outdated word for what we're really talking about. Lineage is the better term, a genetic thread with its own history. Ghost population is how scientists describe something detected only through its genetic aftermath. And the fact that this particular lineage hasn't been found in the fossil record doesn't mean it's fake. It might just mean we haven't dug in the right place yet. Or that we never will. Either way, the DNA insists someone was there. To really appreciate how weird this DNA from a human that doesn't exist idea is, it helps to zoom out and look at the lineages we do know about. The ones that went from bones in the ground to sequences on hard drives, because even those stories were not straightforward. They were resisted, doubted, reinterpreted, and they quietly trained scientists to take ghost lineages seriously. So let's lay down a brutally simplified timeline, roll back to around 700,000 to 800,000 years ago. That's roughly when the ancestors of Neanderthals and Denisovans split off from the lineage that would eventually become modern humans. Before that, you've got earlier species like Homo heidelbergensis, and maybe late Homo erectus wandering around Africa and Eurasia. But those labels blur at the edges and the fossils are patchy. By about 400,000 years ago, you start to see clearly Neanderthal-like skeletons in Europe and Western Asia. Heavy-browed, thick-boned, adapted to cold, with their own stone tool traditions. In Africa, populations on our own branch are evolving in parallel, but they don't look exactly like us yet. In Asia, there are hominins too, but the fossils are sparse and often hard to classify. That emptiness on the map, that's exactly where Denisovans will eventually materialize from almost nothing. Fast forward to about 200,000 to 300,000 years ago. Anatomically modern humans appear in Africa. Faces and skulls that, if you dressed them in modern clothes, would not look out of place walking down a city street. Neanderthals are thriving in Europe and Western Asia. And somewhere in Eastern Eurasia, another branch, closely related to Neanderthals but distinct, is evolving along its own path. We just don't have a name for them yet. Now jump again to somewhere around 60,000 to 70,000 years ago. A small subset of African Homo sapiens leaves the continent, spreads along the coasts and inland into the Middle East, then into Europe, Asia, and eventually Australia. They meet Neanderthals in the Levant and beyond. Later, some of them meet Denisovans in Asia. There are other archaic groups around too, especially in Africa, but those stories are still mostly shadows. For most of the 20th century, that was the broad picture. Neanderthals were the other humans, the side branch that went extinct. We thought modern humans had replaced them, maybe outcompeted them, maybe killed them off. What almost nobody in mainstream paleoanthropology wanted to admit was that we might have blended with them. The first hint that Neanderthals might not be totally gone came from fossils, not DNA. Some modern human remains looked Neanderthal-ish, especially in Europe and the Middle East. But that was easy to shrug off as convergence or just variation. Without genetic proof, the idea of Neanderthals as ancestors, not just neighbours, stayed fringe. That changed in 2010, when a team led by Svante Pääbo and colleagues finally published a draft Neanderthal genome. They had spent years fighting contamination, degradation, and the sheer difficulty of coaxing intact DNA out of bones tens of thousands of years old. When they compared that Neanderthal DNA to people living today, the result was staggering. Non-African populations carried about 1 to 4% Neanderthal ancestry. That number doesn't sound huge, but the pattern was the key. Africans had little or none of that same Neanderthal signal. Europeans, Asians, indigenous Americans, people from Oceania all carried similar Neanderthal-like chunks in roughly the same regions of the genome. The only explanation that made sense was interbreeding. Our ancestors met Neanderthals, had children with them, and those children's descendants survived. It's hard to overstate how disruptive that was. For decades, the replacement model had framed Neanderthals as evolutionary losers pushed aside by the superior Homo sapiens. The genome said, actually, they're still here in your cells. Not as a separate species in a museum case, but as a few % of your immune system, your skin, your brain wiring. That shifted the mental frame from us versus them to us plus them. If Neanderthals were the first big shock, Denisovans were the plot twist nobody saw coming. In 2008, excavations in Denisova cave in Siberia turned up a tiny fragment of finger bone. Not a skull, not a complete skeleton, just a piece of a pinky from a young girl, along with a few teeth. Morphologically, it was almost useless. You couldn't look at it and say, oh, here's a new kind of human. But the collagen was preserved well enough for DNA. When researchers sequenced that DNA and compared it to Neanderthals and modern humans, it didn't fit either. It was clearly closer to Neanderthals than to us, so it belonged on the Neanderthal side of the family, but it was different enough to be its own lineage. That's when the name Denisovan was born, named after the cave, not any anatomical feature. In other words, the genetics defined the species before the bones did. Then came the real mind-bender. When Denisovan DNA was compared to living people, it wasn't Europeans who lit up. It was populations in Asia and especially Oceania. Indigenous Papuan, Australian, and some Philippine groups turned out to have up to 4 to 6% Denisovan ancestry, and traces showed up across East and Southeast Asia as well. These weren't ghosts from a single cave in Siberia. They were a widespread lineage that had spread across half a continent and into distant islands. All of that was reconstructed from a couple of bone fragments and a few teeth. Neanderthals taught scientists that an extinct human could still be inside us. Denisovans taught them that a major human lineage could exist almost entirely as a genetic pattern first, and a handful of ambiguous fossils second. No neat skeleton, no iconic skull, just sequences and statistical comparisons that said, again, someone was here. By the time those results had sunk in, the mental door was open. If we had underestimated how mixed modern humans and Neanderthals were, and completely missed an entire sister lineage like the Denisovans until 2010, what else had we missed? How many other branches had split off, evolved for hundreds of thousands of years, and then vanished physically while their DNA quietly slid into our ancestry? Once you accept that bones are optional and genomes can reveal long-lost relatives, the idea of ghost populations stops sounding like science fiction. It becomes the default expectation, of course, there were more lineages. Of course, some of them left traces only in DNA. The question isn't whether they existed. It's how many, where, and how deeply their stories are woven into our own. And that brings us to the really strange part of this story, how you can go from a handful of genomes, with no matching fossils at all, to the claim that millions of people alive today carry DNA from a human lineage we've never directly seen. To understand that, we have to talk about how ancient DNA is actually pulled out of the past, and why that process is both powerful and treacherous. Ancient DNA starts dying the moment the organism does. Inside a living cell, DNA is constantly being repaired. Enzymes patrol the genome, fixing breaks, patching chemical damage. When the cell dies, that maintenance crew clocks out forever. Water sneaks in and snips the DNA into shorter and shorter fragments. Heat speeds that up. Oxygen attacks bases and changes their chemistry. Microbes move in and chew on whatever is left. Over tens of thousands of years, what used to be long, continuous chromosomes turns into a chaotic soup of microscopic shards. Most of the time, by the time we dig up a fossil, that process is already finished. The DNA is gone, or what's left is so broken and chemically wrecked that it's unreadable. That's why the majority of bones and teeth in museum collections will never give us genomes. Even if they're beautiful fossils, even if they're anatomically important, genetically they're often just empty. And that's before you get to contamination. Your DNA, my DNA, the DNA of everyone who has ever touched that fossil is much fresher, much more abundant, and much more intact than anything left from the original owner. A single skin cell from a modern human carries orders of magnitude more usable DNA than the entire ancient bone. For most of the history of archaeology, people handled fossils with bare hands, stored them in cardboard boxes, washed them, glued them, labelled them with ink. All of that leaves a genetic fingerprint, so when scientists try to extract ancient DNA, they're not just listening for a faint whisper under silence. They're trying to hear a faint whisper under a stadium full of people shouting. To give the ancient signal a chance, labs go almost paranoid. Clean rooms with filtered air, full-body suits, masks, double gloves, surfaces blasted with UV light to break down stray DNA, drills and saws that shave off the outer surface of the bone or tooth, so you're sampling the most protected interior. Even then, some specimens are hopeless. The chemical damage is too severe. The microbial DNA overwhelms everything. You grind, extract, sequence, and you get nothing usable. That's the backdrop to every "we sequenced a Neanderthal" headline. Hundreds of attempts that failed for every one that worked. When you do get DNA out, what actually happens? First, you dissolve the powdered bone or tooth in chemical solutions that pull out any remaining DNA fragments. Then you use enzymes to add little-known sequences to the ends of those fragments, like barcodes and handles. Those handles let you stick the fragments onto a sequencing machine. The machine doesn't care that the DNA is ancient, it just reads whatever letters are present, thousands to millions of fragments at a time. What you get back is not the genome. You get millions of short strings ACTG in different orders, with no obvious labels saying which bit came from which chromosome, or even which species. Mixed into that are microbial sequences, modern human contamination, plant DNA, maybe some cave bear or hyena, if the bone sat in a carnivore den for 40,000 years. The job now is detective work. First, you filter. You align all those tiny fragments against reference genomes, modern human, chimpanzee, sometimes other animals. Fragments that look bacterial get tossed in one bin. Fragments that look humanish go into another. Then you examine the damage patterns. Authentic ancient DNA carries a distinctive scar. Certain bases, especially cytosine, tend to get chemically altered near the ends of fragments over long periods. That leaves a telltale pattern of mutations that modern contamination usually doesn't have. If the human fragments from your Neanderthal bone are short, heavily damaged, and loaded with those terminal changes, that's a good sign they're truly ancient. If they're long, pristine, and look like present-day European DNA, you're probably staring at the fingerprint of the scientists who handled the specimen years ago. Authenticity isn't just one test, it's a stack of clues. Damage patterns, fragment lengths, whether the mitochondrial DNA points to an ancient lineage, whether the nuclear DNA shows combinations of variants that don't match any modern population. The more lines of evidence agree, the more confident you can be that you're looking at the original genome, not someone else's. Even when it works, you almost never get a perfect, complete genome. This is where coverage comes in. Imagine the genome as a huge book. Coverage is how many times, on average, you've read each letter in that book. One-fold coverage means, statistically, you've seen each position once. In reality, some pages have been read 5 times, some not at all, some only partially. 30-fold coverage means you've effectively read the book 30 times over, so you're much more certain about what each letter actually is. Early Neanderthal work had extremely low coverage. Researchers might only have a few % of the genome, or single reads at many positions. That's enough to say, this genome is closer to Neanderthals than to modern humans, or there's evidence of interbreeding. But it's not enough to confidently call every gene or every tiny variant. Later projects pushed some Neanderthal and Denisovan genomes up to much higher coverage, which allowed finer-grained analyses, which genes show signs of adaptation, which regions came from mixing with yet other lineages. Crucially, even partial low-coverage genomes can be incredibly informative. You don't need a perfect book to know which language it's written in. A few scattered sentences already tell you what family it belongs to, and whether it shares paragraphs with other books. That's how a single Denisovan finger bone and a tooth or two could reveal a whole missing branch of our family tree. But there's a catch built into this entire enterprise: sample bias. Ancient DNA survives best in cold, dry, stable environments. Caves are ideal. Constant temperatures, protection from sunlight, often a bit drier than the outside world. Permafrost is even better. At sub-zero temperatures, chemical reactions slow down dramatically, so DNA can hang on for tens or even hundreds of thousands of years. That means our ancient DNA record is heavily skewed towards certain regions and habitats. Europe has lots of limestone caves and a long history of excavation, so Neanderthal DNA is relatively well sampled. Siberia has permafrost and a few key caves, so Denisovans show up there. High, cold places like the Tibetan Plateau preserve surprising signals. By contrast, huge swaths of Africa, South Asia and the tropics are hot, wet and biochemically aggressive. DNA breaks down fast. Bones rot, sediments turn over. Even when fossils exist, they're far less likely to give usable DNA. Add to that political instability, limited funding and restricted access to collections in many regions, and you get a global dataset that is anything but even. When you base evolutionary stories on where DNA happens to survive, you're always at risk of mistaking, "We don't have data for nothing interesting happened here." That's a big reason African ghost lineages are inferred mostly from modern genomes, not ancient bones. Over the last 20 years, though, lab methods have changed what's possible. Early ancient DNA work often failed because scientists tried to treat ancient samples like degraded modern ones. As protocols improved, yields shot up. Researchers learned to focus on the densest parts of bones and teeth, like the petrous portion of the temporal bone in the skull, which can preserve far more DNA. They developed extraction buffers that better pull out ultra-short fragments instead of accidentally throwing them away. They created target enrichment techniques that use synthetic DNA probes to fish out human fragments from a sea of microbial junk, massively boosting the proportion of relevant reads. Sequencing machines themselves became cheaper and more powerful. What once took months and enormous budgets can now be done routinely, allowing multiple attempts on borderline samples and making low-coverage genomes economically feasible. Bioinformatics pipelines got better at distinguishing contamination, modeling damage, and squeezing signal out of noisy data. All of that matters for our mystery, because the more sensitive your methods become, the more you start to see faint, previously invisible signals, tiny shreds of archaic DNA in modern people, or ancient genomes that don't quite match Neanderthals, don't quite match Denisovans, and don't match us. But there's another way to find missing relatives that doesn't rely on any ancient bone surviving at all. You can use living people as your fossil record, and that's where ghost lineages really start to come into focus. If you can't dig the genome out of the ground, you can try to tease it out of the DNA we already have. Think of every person alive today as carrying a compressed archive of their entire ancestry. Not just parents and grandparents, but lineages stretching back hundreds of thousands of years. Most of that history is blurred together, but some pieces are surprisingly crisp. With the right tools, you can look at modern genomes and say, something extra was here. This is where statistical genetics comes in. Instead of starting with an ancient bone and asking, who does this look like? You start with large numbers of living humans and ask, does any part of these genomes look like it came from a population we've never seen? At first glance, that sounds impossible. How can you recognize DNA if you've never seen the original source? The trick is that evolution leaves patterns, and mixing between populations distorts those patterns in distinctive ways. You don't need the missing genome itself. You just need to notice that the pieces we do have don't fit any tree that only includes known branches. One key idea is allele sharing. An allele is just a version of a gene or a DNA site. Maybe at some position in the genome, you can have either an A or a G. Different populations tend to have different frequencies of these versions because of drift, selection, and old splits. If you sample genomes from 3 or 4 groups and tally how often they share the same rare variants, you can start to tell who's more closely related to whom. Under a simple family tree with no interbreeding, those sharing patterns should follow clean, predictable rules. Introduce ancient mixing and the rules break. Suppose you have 3 populations, A, B, and C. Based on everything you know, A and B should be more closely related to each other than either is to C. But when you look at their DNA, you find that A and C consistently share an unusual excess of rare alleles, not just at one site, but across thousands of sites in the genome. You can explain that in 2 ways. Either your whole picture of the tree is wrong, or more plausibly, some ancestors of A picked up DNA from a lineage that was closer to C, and that signal is still visible today. That's admixture, gene flow between branches that had already started to diverge. Now, with Neanderthals and Denisovans, we can check this directly because we have their genomes. But people were already detecting Neanderthal-like signals in modern Eurasians, statistically, before the first decent Neanderthal genome came out. They could see the extra allele sharing was there without knowing exactly what the source looked like. Ghost lineages are the same idea pushed one step further. You see mixing in the statistics, but you don't have any skeleton, any cave, any fossil genome to match it to. Another clue comes from unusual haplotypes. A haplotype is a chunk of DNA where a specific combination of variants tends to travel together, inherited as a block from your ancestors. Recombination shuffles these blocks each generation, chopping them up into smaller and smaller pieces over time. If a population has been isolated for a long time, its haplotypes drift away from everyone else's. When members of that population interbreed with another group, descendants carry some of those distinctive blocks as imports. In modern genomes, you can scan along the chromosomes looking for segments that are very different from the surrounding DNA, internally coherent, like they came as a package, more divergent from other humans than you'd expect from normal variation. Those segments look like they were bolted on from somewhere else. If they're also too diverged to plausibly come from any known archaic group we have data for, then the simplest explanation is there was another branch. It split off early, evolved on its own for a long time, then contributed a small amount of DNA back into the ancestors of some modern population. That's what divergence signals are, statistical hints that some parts of the genome trace back to a lineage that separated long before the main groups we're comparing. The older that split, the more mutations accumulate, and the more those segments stand out as unusually ancient looking. To make sense of all this, scientists build models. They don't just eyeball a weird haplotype and declare new species. They use model-based inference, mathematical frameworks that try out different possible histories and see which ones best explain the observed data. You specify a candidate family tree with branch points, population sizes, and maybe a few episodes of mixing. Then you simulate what the patterns of allele sharing and haplotypes should look like under that history. You compare those expectations to the actual patterns in real genomes. If the fit is bad, you tweak the model, add a pulse of admixture here, change the timing of a split there, adjust population sizes. Sometimes, no matter how you stretch the branches between known groups, you just can't match the real data. There's always leftover sharing that isn't accounted for. At that point, one option is to add a new unsampled branch, a ghost, a population that split off at time X, contributed Y% of its DNA to population Z at time W, but left no direct samples. If including that ghost dramatically improves the match between model and data, and that improvement holds up across different methods and datasets, then you have statistical evidence for a missing lineage. This method is powerful. It let people detect Neanderthal admixture in non-Africans, Denisovan admixture in Papuans, and more subtle mysterious signals in parts of Africa and Asia, all from living genomes. But it can also mislead. Models are only as good as their assumptions. If you misestimate mutation rates, or ignore complex population structure, or leave out certain kinds of migration, the algorithms may solve the mismatch by inventing a ghost branch that isn't real. Sometimes the signal that looks like archaic admixture could actually come from deep structure within ancestral modern human populations that were only partially isolated from each other, not truly separate species. Different demographic scenarios can produce similar statistical footprints. A long-lived cluster of semi-isolated villages trading mates in a complicated pattern can, on paper, look distressingly like a clean split and later hybridization with a distinct archaic group. So when you hear scientists have found evidence of an unknown human species in modern DNA, what they really mean is, our best-fitting models require at least one unsampled, deeply diverged population to explain the patterns we see. It's a strong hint, not a smoking gun. Even so, when independent teams using different statistics, different datasets, and different modeling approaches all keep bumping into the same need for a missing branch, you start to take the ghosts seriously. And some of the strongest, strangest of those signals turn up in exactly the place where the fossil and ancient DNA record is thinnest, Africa. To really see these ghosts, you have to stop thinking of DNA as a list of letters and start thinking of it as a landscape. Across each chromosome, your genome is a patchwork. Some pieces came from one ancestor 5 generations ago, some from another 200 generations ago, some from lineages that split hundreds of thousands of years in the past. The pattern they form, the lengths, the boundaries, the way they're chopped up, that's where introgression leaves its signature. Imagine an ancient mixing event. A small group of modern human ancestors meets a very different population. They have children. Those first hybrid kids carry big, continuous stretches of DNA from each side. Long Neanderthal-like chunks or long ghost chunks alongside the resident genome. Then recombination gets to work. Every generation, when eggs and sperm are formed, chromosomes swap corresponding pieces. It's not random chaos, it's controlled shuffling. Over time, those long introgressed segments are sliced into shorter and shorter fragments, mixed with DNA from all your other ancestors. After a few generations, you can still see relatively long blocks from the incoming population. After hundreds of generations, those blocks are much smaller. After tens of thousands of years, the original huge chunks have been turned into scattered fragments, like confetti. That's why geneticists sometimes talk about recombination as a kind of clock. If you know roughly how often recombination happens per generation, you can use the average length of surviving introgressed segments to estimate when the mixing occurred. Longer segments usually mean more recent gene flow. Shorter, more chopped-up segments point to older events. With Neanderthal DNA in Eurasians, this works beautifully. Most people outside Africa carry about 1–2% Neanderthal ancestry, but not as a single chunk. Instead, it's spread out as many small segments, each a few tens of thousands of bases long scattered across the genome. When you plug those lengths into models of recombination, you get dates for the main Neanderthal-modern human admixture events around 50–60,000 years ago, which lines up with what archaeology and fossils suggest. If you found segments that looked even more diverged than Neanderthal DNA, but they were extremely short and fragmented, that would hint at something older, an introgression event from a more ancient population farther back in time, whose pieces have been recombined to bits. But introgressed DNA doesn't just shrink, it also gets edited. Natural selection doesn't treat all foreign DNA equally. Some of it is harmful in the new genetic background and gets purged. Some of it is neutral and just drifts around. A very small portion might actually be beneficial and get amplified. Think about it from the point of view of a hybrid child 50,000 years ago. If a chunk of archaic DNA messes up fertility, immune function, or brain development, that child will be less likely to leave descendants. Those particular segments disappear from the population over time. This is why, for example, there are deserts of Neanderthal ancestry around certain genes in modern humans, regions where almost no Neanderthal DNA survives, because those variants were selected again. Flip the story, and you get the opposite pattern. Some archaic variants helped with things like altitude tolerance, resistance to local pathogens, or adaptation to cold. Those bits of DNA had an advantage. Individuals carrying them produced more surviving offspring, so those segments spread and sometimes rose to surprisingly high frequencies. The result is a very non-random map. Introgressed segments don't smear uniformly across the genome. They cluster. You see hotspots where archaic ancestry is enriched, places where selection probably favoured those variants, and cold spots where it's almost gone. You also see clustering across populations. Certain groups will share particular archaic segments because they inherited them from the same admixture events, while others don't have them at all. That's one clue researchers look for when they suspect a ghost lineage. If an unusual highly divergent haplotype is found at high frequency in one geographic region, but is rare or absent elsewhere, and it sits in a part of the genome where archaic DNA tends to be tolerated or even helpful, that raises eyebrows. It suggests a local admixture event, followed by selection amplifying that borrowed DNA in that specific environment. But how do you tell those segments apart from normal human variation, especially when you don't have the original archaic genome to compare to? One strategy is to build baselines. Scientists assemble large reference panels of modern genomes representing many populations. They look at how DNA typically varies within and between these groups, and they use that to define what ordinary human diversity looks like. Then they scan along the chromosomes, searching for stretches that are statistical outliers, segments that are much more divergent than typical human segments, internally consistent, as if they came as a block from a single long isolated source, shared unusually between certain populations and not others. For Neanderthals and Denisovans, you can directly match these blocks to the ancient genomes. You literally line up the sequences and see that, say, this 60,000 base fragment in a modern European looks almost identical to Neanderthal DNA from a cave in Croatia, and much more different from typical African sequences. That's clear-cut introgression. For ghost lineages, you don't have that luxury. Instead, you use a combination of tricks. You might ask, if I remove all the regions where Neanderthal and Denisovan ancestry are known, do I still see segments that are unexpectedly ancient-looking? Do I still see haplotypes that sit too far out on the tree, older than the divergence between known groups? You might model how divergent a segment should be if it came from ordinary population structure among early modern humans, versus how divergent it would be if it came from a lineage that split much earlier and then rejoined. If the observed divergence looks too extreme for any plausible just structure scenario that points toward archaic introgression. You also look at coalescent times, the inferred age of the most recent common ancestor for that chunk of DNA. If most of your genome traces back to common ancestors, say 300–600,000 years ago, but a particular segment routinely traces back 1,000,000 years or more, and does so in a way that clusters in certain populations, that's a red flag. It suggests inheritance from a very old, deeply separated branch. Put all of this together, segment length, recombination-based dating patterns of clustering, selection signals, and unusual divergence, and you get something like a fingerprint. Not of an individual, but of an event, a time when a distinct human lineage met our ancestors and left pieces of itself behind. Follow those fingerprints across the world's genomes and a curious pattern emerges. Some of the strangest, oldest-looking fragments don't show up where our ancient DNA record is richest. They show up where we have the least direct evidence, and the most complicated history inside Africa itself. If you zoom out and ask, where are those weird, ancient-looking fragments most common? The answer is not Europe, not Siberia, not the caves where we've pulled Neanderthal and Denisovan bones. It's Africa. And that makes everything harder and more interesting at the same time. First, there's a basic but crucial point. Africa holds the deepest human genetic diversity on the planet. That's not a slogan, it's just what the data say. If you take two random people from, say, Japan and Iceland and compare their genomes, they're different, sure. But take two random people from neighbouring regions in Africa, and on average, they're more different from each other than the Japanese and Icelandic individuals are. You can find more variation within single African countries than you can across entire continents elsewhere. Why? Because our species has been in Africa the longest. More time means more mutations have had a chance to arise and stick around. Populations have split and rejoined, moved with shifting climates, gotten isolated in forests, savannas, mountains and coasts, over and over again. All of that leaves layers of structure in the gene pool. Here's where the headache starts for anyone trying to hunt ghost lineages. The methods that scream introgression in Eurasia were mostly developed in contexts where you had a relatively simple story, a big expansion out of Africa, a few well-timed encounters with Neanderthals and Denisovans, and then spreading out over the rest of the world. You can treat the African source population as your baseline, and anything that looks unusually divergent relative to that baseline, especially outside Africa, becomes a candidate for archaic ancestry. Inside Africa, there is no single clean baseline. There are dozens of baselines layered on top of each other. Imagine a continent where, for hundreds of thousands of years, small groups sometimes get stuck in one ecological pocket, maybe in a forest refugium during a dry phase, or along a river corridor while the surrounding land turns to desert. They accumulate their own mutations, drift away genetically from their neighbours, then later re-expand and mix again when the climate shifts back. From the point of view of the genome, that can look surprisingly similar to having multiple species mixing, even if everyone involved would have recognized each other as humans and could easily have children together. This is what geneticists call ancient population structure. A single species but broken into subgroups that are separated long enough to become quite different, then brought back into contact. Ancient structure can mimic ghost admixture. If you look only at the patterns, the deep divergence of certain segments, their clustering in particular regions, the way they stand out compared to the rest of the genome, it can be hard to tell whether those patterns came from a truly archaic lineage that branched off very early, evolved mostly on its own for hundreds of thousands of years, then mixed back in, or B, an ordinary Homo sapiens subgroup that was just unusually isolated for a long time inside Africa, then reunited with its cousins. Both scenarios can produce very old-looking DNA fragments that are shared mostly within certain African populations. Both can produce coalescent times that stretch back close to 1 million years in some regions of the genome. Both can create haplotypes that sit way out on the edges of the family tree. And right now, we mostly have the patterns, not the context. That hasn't stopped people from trying to tease them apart. Several studies have proposed actual archaic introgression in Africa, real ghost lineages that were as distinct from early Homo sapiens as Neanderthals were, maybe more. Some analyses of West and Central African genomes, for example, have turned up chunks of DNA that look too divergent, too consistently old, to be easily explained by any reasonable model of population structure alone. These fragments sometimes show up at modest frequencies, are highly unusual compared to other African sequences, and appear to trace back to ancestors who split off perhaps 600,000 to over 1 million years ago. Those papers suggest that our African ancestors may have interbred with deeply divergent hominin groups, local archaics, that never left much of a fossil record, but left descendants in our genomes. In some models, multiple archaic lineages are invoked, maybe a ghost cousin mixing with ancestors of certain West African groups, another with ancestors of some hunter-gatherer populations, each at different times. Other researchers are more cautious. They point out that if Africa has been home to many semi-isolated Homo sapiens populations for hundreds of thousands of years, and if those populations sometimes bottlenecked, sometimes expanded, sometimes swapped migrants in lopsided ways, you can get patterns that look extreme without needing to invoke a separate species. Change the timing of splits, the duration of isolation, the sizes of those groups, and the ghost signal can often be reproduced in simulations using nothing but structured Homo sapiens ancestors. In that view, what we're seeing in African genomes are the ripples of a very complicated, very old population tree, not necessarily the fingerprints of completely separate hominin lineages. So, which is it? This is where Africa's biggest strength, its deep history, becomes our biggest limitation. We have almost no ancient DNA from there. In Europe and Siberia, you can literally pull Neanderthal or Denisovan DNA out of bones and dirt and line it up with modern genomes. That lets you say, yes, this weird-looking chunk in present-day people matches that specific ancient population. You can anchor your inferences in something concrete. In most of Africa, the climate is brutal on DNA. Heat, humidity, microbes, over tens of thousands of years, they chew genetic material to pieces. There are a few successes from more recent time periods, but nothing like the older, high-quality Neanderthal and Denisovan genomes we rely on elsewhere. Without those ancient African genomes, we're trying to read a palimpsest, a text that has been written over many times, where faint traces of earlier writing are still there but hard to separate. We see the unusual segments, we can measure their age and divergence, but we don't know whether they belong to a truly separate, long-split hominin lineage that never got a name, several such lineages, each contributing a bit, or a tangled network of Homo sapiens subgroups that were just very, very structured for a very long time. Most likely, the answer is not all or nothing. Africa was large enough, and old enough, for both things to have happened, deep structure within our species, and encounters with more archaic neighbours. For now, though, the continent that holds the oldest chapters of our story also holds the biggest blind spot. The strongest hints of a ghost lineage show up right where our direct fossil and DNA evidence is thinnest, which means the most dramatic claims about unknown African humans are also the hardest to test. And while Africa keeps its secrets, the picture outside the continent has become surprisingly clear. Once our ancestors left, they didn't just meet Neanderthals and Denisovans. The ghost pattern pops up there too, just in a different, slightly less tangled form. Outside Africa, you'd think things would be simple. Cool, dry caves, permafrost, bones that actually hang on to DNA for tens of thousands of years. Europe and Northern Asia are the places where ancient DNA really took off as a field. This is where we got the first Neanderthal genome, the Denisovan fingerbone, a parade of Ice Age hunter-gatherers whose genomes we can sequence almost like living people. If anywhere on Earth should be solved, at least at a coarse level, it should be Eurasia. And yet, even here, the genomes won't quite line up with the neat story we'd like to tell. When geneticists first started comparing modern Eurasian genomes to Neanderthal DNA, the pattern looked relatively tidy. Non-Africans had about 2% Neanderthal ancestry. Some populations in Oceania had an extra 3–5% of something else, which later turned out to match Denisovan DNA. You could draw a picture. Modern humans leave Africa, bump into Neanderthals in the Middle East, pick up some genes, then later bump into Denisovans somewhere in Asia, pick up a little more. But as datasets got bigger and methods got sharper, that picture began to fracture. One of the first cracks was the realization that Neanderthal ancestry in Eurasians doesn't come from a single rendezvous. It comes from multiple pulses of gene flow at different times in different places. East Asians, for example, tend to carry slightly more Neanderthal ancestry than Europeans, and not all of that extra fraction matches the Neanderthals we've sequenced from Europe. Some chunks in modern East Asian genomes look like they came from a Neanderthal population that had already split away from the Neanderthals who left bones in Croatia or Spain. The same is true for some Siberian and Native American ancestries. You can see little signatures of at least 2, maybe 3, distinct episodes of Neanderthal admixture involving somewhat different Neanderthal groups. Now add Denisovans. At first, Denisovan DNA looked like a clean third branch story. You had Neanderthals in the west, Denisovans in the east, and modern humans coming up in the middle. But when you start comparing the Denisovan fragments in modern peoples, they don't all resemble that single Denisovan genome from Denisova Cave in Siberia. Some Denisovan-like segments in Papuans and Aboriginal Australians cluster with the Siberian Denisovan. Others sit off at a noticeable distance, as if they came from a cousin lineage that had already split from the Denisovans in that cave. Populations in East Asia show yet another mixture, some fragments that look close to Neanderthals, some that are clearly Denisovan-like, and a few that are somewhere in between, or off to the side. By the time you've mapped all of this, Denisovan starts to look less like a single species and more like a catch-all name for several deeply structured lineages spread across Asia. In other words, there may have been Denisovans of the Siberian mountains, Denisovans of mainland Southeast Asia, Denisovans of island chains stretching toward New Guinea, each diverged from the others for hundreds of thousands of years, each meeting different waves of modern humans at different times. And that's just the pieces we can anchor to known reference genomes. When you look across Eurasia more broadly, there are segments of DNA in modern people that are clearly archaic in flavour. Old divergent, low recombination breakpoints, all the hallmarks of introgression. But they don't fit Neanderthal or Denisovan profiles very well. They're too different from African Homo sapiens to be just ordinary variation. But when you try to assign them to Neanderthals or to Denisovans, the match is poor. It's like having a fingerprint from a crime scene that definitely isn't yours, but doesn't quite match any suspect in the database either. Some of these odd fragments show up in East Asians and Oceanians. Others appear at low frequency in Europeans or in Siberian groups. In some analyses, they show patterns of allele sharing that hint at a donor population that split from the Neanderthal Denisovan line very early, then had brief contact with the ancestors of particular modern human groups before vanishing. That's where the phrase ghosts in Eurasia really starts to earn its name. The tricky part is, how do you distinguish between we're missing some Denisovan subgroups and we're looking at a truly unknown lineage, separate from both Neanderthals and Denisovans? Because once you admit that Denisovan probably hides multiple long-separated populations, some of those weird signals could just be unrepresented Denisovan diversity. We only have a handful of Denisovan genomes, from one cave and a couple of teeth and bones. It would be like trying to describe all of modern human diversity from 2 skeletons dug up in Norway. So geneticists set a high bar before they're willing to say, this is beyond Neanderthal and Denisovan. Strong evidence for a truly unknown Eurasian lineage would look something like this. First, the divergent fragments in modern genomes would consistently fall outside the variation seen in any sequenced Neanderthal or Denisovan, even after you model in plausible unsampled cousins of those groups. Statistically, you'd want a clear 4th cluster on the tree, not just a fuzzy edge of an existing branch. Second, the introgressed chunks would hang together across individuals in a patterned way. You'd expect to see blocks of DNA that co-occur with shared ancestry dates in multiple populations. That would suggest a real historical population that mixed with modern humans, not just random outliers or artifacts. Third, the timing would need to be coherent. Using recombination and mutation rates, you can estimate when the gene flow happened. If those dates form a cluster, say pointing to an admixture event 150,000 to 200,000 years ago in Southeast Asia, that's more convincing than a smearing of ages that could just be background structure. Fourth, the signal should survive ruthless attempts to explain it away. You'd throw every alternative at it. Complex population structure inside Neanderthals, multiple Denisovan sublineages, back migration from one region to another, strange demographic histories in modern humans. If the ghost signal still stands out after all that modelling, then it starts to look real. And finally, you'd want convergence from different kinds of data. Not just one method, not just one lab. Ideally, several independent analyses using different approaches, haplotype-based scans, site frequency methods, coalescent modelling, would all point to the same ghost. In some parts of Eurasia, we're close to this threshold. In others, what we call mystery may just be our incomplete sampling of Neanderthals and Denisovans stretched across a vast continent and hundreds of thousands of years. But the fact that any of these questions are still open in the best sampled half of the planet tells you something important. Even with dozens of ancient genomes in hand, even with caves that act like natural freezers for DNA, the story of who we met and who we mixed with is still missing characters, and that uncertainty becomes critical when you go back to a single moment in time, when modern humans first walked into a world already occupied by other humans. The Denisovan story is the perfect cautionary tale for all of this, because Denisovans were, in a sense, a ghost lineage before we even realised how big their shadow was. They didn't burst onto the scene with a spectacular skeleton or a beautifully preserved skull. They showed up as a tiny piece of bone so nondescript that the original excavators didn't even know it was human. In the early 2000s, a team digging in Denisova Cave in the Altai Mountains of Siberia found a little fragment from the end of a finger. No distinctive brow ridges, no jaw, no teeth you could compare to Neanderthals or early Homo sapiens. Just a sliver. It went into a box. Nobody thought, Ah yes, new human species right here. Years later, when ancient DNA methods had improved enough to handle very degraded material, that little fragment was chosen for sequencing, more as a test than anything else. Can we get DNA out of this? Does it match Neanderthals, or modern humans, or nothing at all? When the genome came back, it was a problem. It clearly wasn't modern human. It clearly wasn't a Neanderthal either. It was related to Neanderthals but split off earlier, forming its own distinct branch of the archaic human family. Genetically, it sat somewhere between Neanderthals and the common ancestor we all share. The team realized they were looking at a lineage of humans we had never seen in the fossil record, a whole branch of our family tree, known only from one child's finger. They named it after the cave, Denisova, Denisovans. At that point, the story could have stayed small, a quirky footnote. Strange mountain cousins of the Neanderthals, known from a single bone and a tooth. But then geneticists did the obvious follow-up. They compared the Denisovan genome to living people. And suddenly the map of the world lit up. Populations in Melanesia, Papua New Guinea, parts of Indonesia, Aboriginal Australians, they weren't carrying a tiny trace of Denisovan ancestry. They had multiple percent. More Denisovan ancestry than most non-Africans have Neanderthal. This wasn't a minor encounter. This was substantial mixing with a large, successful population that had to be widespread across Asia and the Pacific. Then researchers checked East Asians, some groups in China, Tibet, mainland Southeast Asia. They had Denisovan ancestry too. Not as much as Papuans, but enough to be unmistakable. Remember, this was all being discovered when our fossil evidence for Denisovans was still essentially that one finger, a tooth and some bone fragments. Maybe a mandible from the Tibetan plateau, tentatively linked later. That was it. From a handful of bones, you get the genetic fingerprint of a human group. From that fingerprint, you discover that millions of living people carry their DNA. The order of discovery was backwards. We didn't map their fossils, track their migration, and then check genomes. We found the DNA first, and only later realized how far that DNA had travelled. And as researchers dug deeper into those Denisovan-like fragments in modern genomes, it got even messier. The Denisovan ancestry in Papuans, for example, doesn't all point to the same source. Some segments look closely related to the Denisovan from Denisova Cave. Others look like they came from a population that had already been separated from the cave Denisovans for hundreds of thousands of years. East Asians tell a similar story, but with a different mix. Some of their archaic fragments match the Siberian Denisovan. Some look more distant. Some blur toward Neanderthal territory. It's like opening a drawer labelled Denisovan and finding several different sets of house keys inside, each to a different city. That led to a new picture. Denisovan stopped meaning one uniform, coherent population. Instead, it started to look like an umbrella term for multiple, long-separated branches of an Eastern archaic lineage. Denisovans of the Altai, Denisovans of the Tibetan Plateau, Denisovans of island Southeast Asia. Each branch diverged in deep time. Each developed its own history, its own adaptations, its own points of contact with different waves of Homo sapiens. Some groups met our ancestors on the Asian mainland. Others met them on stepping stone islands in Wallacea. Still others may never have met modern humans at all before vanishing. But in our vocabulary, they all got collapsed into one name, Denisovan. And this is where the lesson kicks in. If you looked only at the first Denisovan genome, you might think you're dealing with a single population, a neat third branch alongside Neanderthals and us. One species, one story, one set of traits. Once you add modern genomes into the mix, that simplicity evaporates. You see branching within the branch, structure inside the label, genetic distance is big enough that if you had the bones, paleoanthropologists might be arguing whether some of these Denisovans were actually separate species. So when scientists today talk about unknown archaic ancestry in Africa, or in parts of Eurasia, the Denisovan case is always sitting in the background, because it reminds us of a few uncomfortable truths. First, you can discover an entire human lineage from almost no bones at all. The quality of the DNA matters more than the completeness of the skeleton. Second, a single reference genome does not define the full diversity of a group. One Denisovan from one cave does not speak for all Denisovans across half a continent. When we say matches Denisovan, we really mean matches this tiny sample of Denisovans we happen to have sequenced. Third, a name is just a label we slap on a piece of a much more continuous reality. Nature doesn't draw a bright line between Denisovan A and Denisovan B, or between late Denisovan and early unknown ghost. Populations split, drift, merge and split again. Our categories try to keep up, but they're always a bit behind. So when we pick up a strange signal in modern DNA, something that doesn't quite match Neanderthals, doesn't quite match Denisovans, and doesn't look like ordinary modern human variation, we have to be careful how quickly we call it a new species. Is it truly a fourth branch, some long-lost cousin we've never sampled? Or is it another Denisovan-like group that we just haven't met in the fossil record yet? Another branch hiding under the same umbrella label. The Denisovan lesson is simple but powerful. The first time you give a name to a lineage is almost never the moment you understand its reach. And that lesson is exactly what we have to keep in mind when we start talking about something even stranger than Denisovans. A human lineage that, as far as we can tell, doesn't exist in any cave or museum drawer, but still leaves fingerprints in the DNA of millions of living people. So how do you even test a claim like that? How do you go from a vague hunch, this DNA looks weird, to a defensible statement like, something very Neanderthalish but not Neanderthal, mixed with the ancestors of these people roughly this many years ago? The short answer is, with a set of tools that sound terrifying if you see the equations, but are actually based on a couple of simple ideas. The first idea is this. If two populations shared a recent ancestor, they should share more genetic variants with each other than with a more distant group. The second idea, if there was mixing, if one population picked up DNA from a third archaic group, that sharing pattern gets skewed in very specific, detectable ways. The most famous test built on this is something called a D-statistic. People also call it Abba-Baba, which sounds like baby talk, but is actually about counting patterns. In practice, you take 4 genomes, imagine them as 4 lineages, chimpanzee as an outgroup, then 3 human populations, say a West African group, a European group, and Neanderthals. At a particular spot in the genome, there are 2 possible letters you care about. Call them A and B. You ask, who matches whom? If our tidy family tree was right, no mixing, just clean splits, you'd expect the number of sites where Europeans and Neanderthals share the same letter to be about the same as the number where West Africans and Neanderthals share it, once you control for their place on the tree. But if Europeans have Neanderthal ancestry, and West Africans largely don't, then Europeans will match Neanderthals more often than expected. Those extra shared sites show up as an imbalance in Abba versus Baba patterns when you add up millions of positions across the genome. The math behind D-statistics is a bit gnarly. The concept isn't. It's basically, do we see more shared variants between these two than we should, given the tree we think is true? If the answer is yes, and it's yes in a big, statistically convincing way, that's one line of evidence for gene flow. That's how the first strong claim of Neanderthal introgression into non-Africans was made. It's also how people started noticing that some African populations share excess alleles with unknown archaic branches that don't look like Neanderthals or Denisovans at all. But that's just one test. You don't want to bet everything on a single statistic. So researchers move up a level and start drawing what are called admixture graphs. If a normal evolutionary tree is a set of branches that only split, an admixture graph is a tree that's allowed to reconnect some of those branches with little arrows, mixing events. On these graphs, each node is a population, and each line represents ancestry. You start with something simple, a clean branching tree where a common ancestor splits into different modern groups and archaic groups. Then you ask, does this simple tree reproduce all the observed allele sharing patterns? Usually it doesn't. The real data show that, say, Oceanians share too much with Denisovans, or that some West African groups share too much with a ghost branch that's neither Neanderthal nor Denisovan. So you start adding edges. What if we allow 3% of the ancestry of this population to come from this archaic branch? You fit the model. You see if the mismatch between the graph and the real data goes down. The process is iterative, and a bit like forensic reconstruction. Try a tree, measure the error, add a mixing edge, see if it gets better. Move the edge, test again. The final best fit graph is not a perfect map of reality, but it gives you a structured, testable story. Which populations split when, and where the big pulses of archaic introgression likely happened. These statistics and admixture graphs work at the population level, broad patterns of who shares what with whom. But there's another family of tools that zoom in much closer, local ancestry methods. Instead of just asking, does this population have archaic ancestry? These methods scan each person's genome, chunk by chunk, trying to flag individual segments that look unusually archaic like. The logic is this. Most of your genome looks like a jumble of modern human variation. But if your ancestors ever mixed with Neanderthals or Denisovans or some unknown cousin, then small stretches of DNA from that archaic group can persist for hundreds of thousands of years, recombining and shrinking each generation, but still recognizable. Those archaic-like tracts tend to be a bit more divergent from the modern human reference, more similar in their overall shape to known archaic genomes if you have them, clustered in lengths that match what you'd expect from a mixing event at a particular time in the past. Algorithms like Strutus or newer machine learning approaches slide along the genome, measuring patterns of variation in windows. Where they see something too old, too odd, and too similar to a known archaic signature, or too archaic in general, even without a perfect reference, they flag it. That's how people can say things like, on average, Papuans have this many percent Denisovan ancestry spread across thousands of little tracts, or these African genomes contain segments that look like they're from a lineage that split from the main human line more than 1 million years ago. But even that isn't the end of the toolkit. Behind all of these methods is a quieter, less glamorous piece: simulation and model comparison. You might detect an archaic-looking signal, but you still have to ask, could a messy but ordinary history produce this without invoking a whole new lineage? To test that, researchers simulate genomes under different scenarios. Scenario A: a simple tree with no ghost introgression, but with realistic population size changes and structure. Scenario B: same as A, but add 5% admixture from a deeply diverged ghost population into one branch, 50,000 years ago. Scenario C: 2 ghost events, one into this branch, one into that one. For each scenario, you generate fake genomes and run the same statistics and local ancestry methods you used on the real data. Then you compare. Which scenario reproduces the weird allele sharing patterns? Which one reproduces the length and frequency of archaic-like tracts? Which one matches the overall distribution of genetic diversity? This is where ghost lineages either survive or die. If the no-ghost model can't reproduce the data, but the ghost introgression model can, over and over again across multiple tests, the ghost explanation starts looking less like a spooky story and more like the simplest working hypothesis. Of course, different tools don't always agree. A D statistic might scream introgression, while an admixture graph can sort of fit the same pattern by tweaking branch lengths instead. Local ancestry methods might find a lot of archaic-like fragments in one region, but they're borderline in another. One simulation framework might favour an early ghost event, another might fit a later one. This is why you almost never see serious researchers declare, "We have discovered a new human species hiding in your DNA," based on a single analysis. Instead, they layer methods. If D statistics admixture graphs, local ancestry scans, and simulations all independently converge on a similar story, an extra branch, splitting this deep in time, contributing roughly this much to these populations, that's when people start talking seriously about unknown archaic ancestry. And when the tools point in slightly different directions, that's when the real work begins. Tuning the models, questioning the assumptions, asking whether the signal could be coming from Denisovan, like diversity we haven't yet sampled, or from complicated structures within early Homo sapiens themselves. Because before we can claim a ghost, we have to rule out every ordinary explanation we can think of. And that's where things get contentious, because some of those ordinary explanations can be surprisingly good at imitating ghosts. So let's walk through the main ways you can get a ghost DNA signal without actually having a ghost. The first big one is ancient population structure. We tend to talk about early humans in Africa as if they were one big panmictic blob, everybody happily mating with everybody else. That was almost certainly wrong. Imagine instead that Africa 300,000 or 500,000 years ago was already full of semi-isolated populations. Forest dwellers here, savanna specialists there, groups hugging coastlines or big river systems, some exchanging partners regularly, others cut off for long stretches by deserts, mountains or climate swings. If those groups split from each other, stayed apart for a long time, and only partially mixed back together later, you get lineages inside Homo sapiens that are very old and very weird looking in genetic space. Weird enough that when you compare them to a simple tree, they can look like DNA from a separate archaic species. You run your D statistics, and one African population seems to share an excess of alleles with a very deep branch. Your admixture graph happily draws a ghost arrow, 5% from an unknown archaic lineage that diverged 900,000 years ago. But you might not need that extra branch at all. A long period of structure, multiple Homo sapiens-ish groups diverging and reconnecting at different times, can create the same lopsided sharing patterns you'd expect from introgression. It's like mistaking a very old, very tangled family tree for a marriage between two completely different families. And we know Africa had structure. Fossils, stone tools, climate models, and modern genetic diversity all point to a continent that was more like a patchwork of semi-connected populations than a single unified group. So one constant question is, are we really seeing gene flow from a separate ghost species, or just the genetic echoes of an already structured, diverse human population? The second big confounder is reference bias. All of these tests rely on comparison. Archaic-like only means different from our current panel of modern humans in a way that reminds us of Neanderthals or Denisovans, or of something that split off even earlier. But what if our panel is missing a lot? Right now our archaic references are a handful of Neanderthals, mostly from Europe and Western Asia, and a few Denisovan fragments from one Siberian cave, plus some lower coverage bits from Tibet and China. That's like trying to reconstruct African elephant evolution with DNA, from 3 zoo animals and 1 mummified tusk. If the real Denisovan family was very diverse, multiple branches spread across Asia, and we've only sequenced one corner of it, then any DNA that's Denisovan-ish, but not close to our Siberian Denisovan, can look like it comes from a totally different lineage. The same problem hits our modern reference panels. Most genome projects oversampled certain groups: Europeans, East Asians, a few big city populations. Many deep, rural or formerly isolated communities are still underrepresented. So, modern human variation in our databases is narrower than the variation that actually existed. Put those together and you get a dangerous combo: an incomplete archaic reference and an incomplete modern reference. Now segments that are just unsampled Denisovan or unsampled early African diversity can masquerade as completely unknown ghost DNA. This is why adding more genomes can literally erase ghosts. A signal that looked like a mysterious archaic branch in 2012 can soften into, oh, that's just this unsampled modern group, once you finally sequence that group and add it to the comparison. Then there are bottlenecks, drift, and sampling quirks. Genetic drift is random change. Bottlenecks, a few hundred or a few thousand people founding a new population, turbocharge it. Think of it as rolling the dice on which variants get carried forward. If a small group leaves Africa, passes through a harsh bottleneck, and then explodes in size, its descendants can end up with odd patterns of allele frequencies that don't match simple expectations. Certain rare ancient variants might be unusually common just because they got lucky in a tiny founder group. When you later compare that population to others, the statistics might scream archaic introgression, simply because the mix of old and new variants is skewed in a way that looks like a pulse of foreign DNA. Uneven sampling makes this worse. If you happen to sequence 5 genomes from a village that went through its own local bottleneck, or from a group with lots of internal relatedness, you're effectively listening to a very small, quirky sample and pretending it stands for millions. Any local oddity can get amplified into a global signal. Next, method sensitivity and overfitting. Admixture graphs in particular are seductive. You start with a tree that doesn't fit. You add one migration edge and the fit improves. You add another and it improves again. With enough arrows, you can make almost any messy pattern fit almost perfectly. At some point, you're not discovering history anymore. You're curve fitting. Different software packages, or even different starting assumptions with the same software, can spit out different best graphs. One with a ghost pulse here, another with slightly longer branches and no ghost at all. Both might fit the data about equally well. Local ancestry methods have their own sensitivities. Change the threshold for archaic-like just a bit, or train your machine learning model on a slightly different set of reference genomes, and the number of ghost candidate segments can jump or collapse. This is why responsible researchers obsess over robustness. They don't just ask, can I fit the data with a ghost? They also ask, can I fit the data without one? If I push the ordinary parameters, structure, drift, variable population size, as far as they reasonably go. And that brings us to the last filter, replication across datasets. A fragile ghost appears only in one study, using one method on one narrow dataset. Change the population, update the reference genomes, or tweak the assumptions and the signal evaporates. A sturdy ghost survives abuse. It shows up in whole genome data and in targeted SNP arrays. It appears when you analyze West Africans this way, and again when you analyze a completely different African group with a different tool. It's there when you use Neanderthals and Denisovans as references, and it's still there, maybe a bit weaker, but recognizably there, when you deliberately leave those references out. You want converging lines of evidence, D statistics that stay significant under different configurations, admixture graphs that keep asking for an extra branch, local ancestry scans that keep flagging deep, divergent tracts in roughly the same places, simulations that consistently fail to reproduce the patterns without some form of archaic introgression. Only when a signal survives that gauntlet do people start saying, okay, this might not just be population structure or weird drift or a missing sample. This might actually be DNA from a lineage we haven't seen yet. And if that's true, the next question is irresistible. What kind of humans were they? To answer that, we have to start with something humbling. DNA is both incredibly powerful and incredibly limited. From genetics alone, we can say some things with decent confidence. We can estimate how long ago this lineage split from ours. We can see whether their DNA carries signs of adapting to heat, cold, altitude, diet, disease. We can see if they had slightly different immune systems, different metabolic quirks, maybe even different sleep patterns. But we can't reconstruct their faces. Not really. Those glossy, this is what a Denisovan looked like reconstructions you see? They're mostly educated guesswork built from a few skeletal fragments, comparisons with Neanderthals and modern humans, and a lot of artistic license. For a lineage we only know from ghost DNA, we don't even have that. We won't know their exact skin colour, hair, texture, eye shape, the way they walked or the way their voices sounded. At best we can say things like, their pigmentation genes look similar to early Homo sapiens in this region, or they probably had darker skin because these variants are associated with UV protection in strong sunlight. Behaviour is even trickier. There is no gene for culture, no single mutation for they were peaceful, or they were violent, or they were shamans. What we can occasionally see are statistical hints, segments of archaic like DNA in modern humans that affect, say, brain development, synapse formation, or hormone regulation. We can see that those segments spread or were pruned away by natural selection. If a ghost lineage contributed a bunch of variants that affect, for example, our circadian rhythm, or how we respond to hypoxia, or how easily we store fat, that tells us something about the challenges they were facing. It tells us what kind of problems their bodies, and probably their societies, had to solve. So we're not painting a portrait, we're inferring a lifestyle from footprints. One of the clearest clues is divergence time. When geneticists say this ghost lineage is deeply diverged, they're talking about when its ancestors split off from the ancestors of the populations we already know. Some estimates for African ghost lineages land in the range of 500,000 to over 1 million years ago. Others are shallower, maybe 300,000 or 400,000 years, and every estimate comes with big error bars and lots of caveats. But suppose one of these lineages really did branch off around, say, 700,000 to 900,000 years ago, around the same time as the ancestors of Neanderthals and Denisovans. That would mean it was not just an unusual Homo sapiens group. It would have had hundreds of thousands of years to wander down its own evolutionary path. Different climates, different predators, different diseases, different ways of finding food, competing, cooperating. Given that kind of time, even if the bodies still looked broadly human, the details could have been strikingly different. Skull shape, body proportions, voice range, maybe developmental timing, how long childhood lasted when puberty hit, how long they lived. On the other hand, some ghost signals might come from much more recent splits, old branches within Homo sapiens or near sapiens populations that were semi-isolated for tens of thousands of years, not hundreds of thousands. Those would have looked and behaved a lot more like other early modern humans, just with their own local twists. Where could these people have actually lived? To leave DNA in us, they had to meet our ancestors at some contact zone. That narrows things down. For African ghost lineages, likely candidates are ecological boundaries and refuges. forest savanna edges in Central or West Africa, river corridors that stayed green during dry phases, or coastal refugia along the Atlantic or Indian Ocean, where human populations could hang on while the interior became harsh. Imagine a lineage that specialized in dense forests, shorter travel ranges, different hunting strategies, more reliance on plant foods, maybe more knowledge of toxins and medicines. Meanwhile, a neighbouring sapiens-ish population thrived in open savannas. For most of prehistory they might barely see each other. Then climates shift, forests shrink or expand, and their territories overlap. A few mixed children survive. Ghost DNA slides quietly into our lineage. Outside Africa, some of the extra archaic-like signals in Eurasia might point to ghost relatives of Denisovans or Neanderthals that lived in places where fossils preserve poorly: tropical Southeast Asia, parts of South Asia, humid lowland forests where bone dissolves fast. If there was a deeply diverged Asian lineage that liked warm, wet environments, built boats, and hugged coastlines and islands, it could have interacted with early modern humans moving along those same coasts, and left almost no skeletons we can find today. Which brings us to the fossil record. Why might it have missed them? First, fossils are rare. Most bodies never fossilize. They rot, get eaten, get scattered. Then you need just the right conditions: rapid burial, low oxygen, the right minerals. Then after all that you need erosion to expose them, and a human, in the right time, walking over that exact patch of ground, who recognizes that funny-shaped rock as a bone. Second, even when we do find fossils, identification is messy. Skulls and bones don't come with barcodes. A jaw from 400,000 years ago might look sort of like early Homo sapiens, sort of like something more archaic, and end up either in a drawer or in a category like early Homo or Homo heidelbergensis that quietly lumps together multiple lineages. If an unknown lineage looked intermediate, partway between known forms, it could just get folded into an existing species. That's almost certainly happened already. Some of our known species may actually be mosaics of several ghost populations. And there's another wrinkle. We might not be dealing with just one unknown lineage. The DNA patterns we see could be the blended echoes of multiple ghosts, several deeply diverged groups in different regions, each contributing a little at different times. When all that gets stirred together for hundreds of thousands of years, our statistical tools struggle to untangle 1-8% pulse from lineage X versus 3% pulses from slightly different lineages that were already related to each other. So, in one model, there's a single, relatively coherent unknown species, long separated, partially isolated, that occasionally meets and mixes with our ancestors. In another model, there's a shadow ecosystem of archaic humans, forest specialists, highland specialists, river valley foragers, island hoppers. Some are closer to us, some more distant, some more like Denisovans, some more like early African Homo. Each leaves a faint genetic fingerprint that overlaps with the others until, from our vantage point, it just looks like a deep, strange ghost branch. Right now, the data don't let us choose cleanly between those pictures. What we can say is this: if these ghosts were real populations, they were humans in the broad sense. Upright, tool-using, fire-managing apes, with social lives and skills and traditions, trying to get through ice ages and droughts just like our direct ancestors were. And somehow, pieces of them are still here, scattered through the genomes of millions of living people, carried silently in cells that divide, in children who are born, in bodies that adapt to heat, to parasites, to thin air, using solutions first hammered out by a lineage we've never seen. Which raises the next question: how did DNA from a small, possibly marginal population end up spread across entire continents? Because ancestry doesn't spread like paint on a wall. It spreads like gossip in a small town. You don't need a huge population to leave a massive genetic footprint. You just need the right people in the right place, at the right time, and a lot of babies. Imagine a tiny ghost population living in one valley. A few hundred individuals, maybe fewer, for tens of thousands of years. Nobody outside that region ever meets them. Then climate shifts or rivers change course, or somebody follows a herd too far. One day, a small band of early Homo sapiens-ish humans wanders into their territory. What happens next doesn't have to be a dramatic invasion. It can be something as simple as a handful of mixed families. A few ghost lineage individuals join the incoming group, or vice versa. Maybe there's conflict first, then truce, then trade, then marriages. Over a couple of generations, you get children with half-ghost ancestry. Their children have a quarter, their grandchildren have an eighth. Genetically, the ghost DNA is already being diluted. But here's the key. The people carrying it might be part of a population that's about to explode in size. This is where founder effects come in. A founder effect is what happens when a small subset of a population goes off and starts a new branch. Think of a few dozen people settling a new island, or crossing a narrow sea, or moving into a freshly opened corridor of habitable land after an ice sheet retreats. Whatever genetic quirks they happen to carry, by luck, not planning, get baked in as the starting recipe for that new population. If those founders happen to carry ghost DNA, that ghost ancestry doesn't stay rare. As the group grows, it gets amplified. It goes from a weird little local signal in a few families to a background ingredient in everyone's genome. Do that a few times over tens of thousands of years, and you can get a situation where a very small, very local lineage ends up with descendants spread from one end of a continent to the other. This is exactly why ancestry can be common even if the original source population was tiny. They didn't have to conquer the world. They just had to mix with populations that later did. Think about the out-of-Africa expansion of modern humans around 60–70,000 years ago. Genetic studies suggest that all non-African humans alive today may trace back to a surprisingly small founding population, maybe just a few thousand breeding individuals, possibly fewer. That's an extreme founder effect. It means any DNA those founders carried, even rare variants, had a shot at riding along as humanity colonized Eurasia, Australia, and eventually the Americas. If some of those founders already had bits of Neanderthal DNA, or Denisovan DNA, or ghost DNA from an even older lineage, those archaic fragments hitched a ride on the greatest population expansion in human history. You see a similar pattern on a smaller scale within regions. A small group develops some innovation, better tools, better social organization, maybe a particularly favorable ecological niche. They grow faster than neighboring groups. Over time, they send off splinter bands, each one carrying a shuffled sample of their genetic deck. Some of those spin-off groups replace or absorb other populations. Others just blend, creating hybrid communities whose descendants keep carrying mixtures of ancestries further and further away from the original contact zone. After enough cycles of this, ancestry spreads in a way that's completely out of proportion to the size of the founding group. But there's an important distinction here. Having any ghost ancestry versus having a lot of it. You can think of your genome as a giant library of about 3 billion letters. Archaic DNA, Neanderthal, Denisovan, or ghost, is like a handful of shelves scattered through that library. Almost everyone outside Africa today has some Neanderthal DNA. That doesn't mean they have the same amount or the same pieces, or that Neanderthal ancestry dominates their genome. It's usually a few percent at most, and that percentage itself is an average over many tiny fragments. Over thousands of generations, recombination shuffles and slices ancestral DNA into shorter and shorter pieces. Some pieces get lost entirely by chance, others are kept, or even boosted, if they help survival in some environment. So when geneticists say millions of people have DNA from an unknown lineage, that can mean most of those people have a few small segments here and there that look like they came from a very old branch. Not that they're 20% ghost. And here's where things get even more counterintuitive. Genealogical ancestry versus genetic ancestry. Genealogically, your family tree doubles every generation you go back. 2 parents, 4 grandparents, 8 great-grandparents, 16, 32, 64, and so on. Go back 1,000 years, and the number of slots in your tree is ridiculous. Many more than the number of people who are actually alive in your region. Lineages overlap. You get the same ancestors showing up in multiple branches. That's how you can be descended from thousands of people whose DNA didn't all make it into your genome. Genetically, you only inherit half your DNA from each parent, and recombination shuffles which half. After enough generations, you start to lose entire ancestors at the DNA level. They're still your ancestors in the family tree sense, but you might carry 0 identifiable DNA from them. Push this back tens of thousands of years and the pattern gets extreme. You can be genealogically descended from hundreds or thousands of individuals in an archaic or ghost population, while carrying only a handful of tiny DNA fragments from them. Flip it around, and the reverse is also true. A few individuals can end up ancestors to an enormous number of people. If a small mixed community happens to be in the right place, on a migration route, at a fertile river junction, or in a refuge that supported rapid growth, that group can contribute ancestors to vast swaths of the future population. Those few archaic admixed individuals might not have looked or felt special at all in their own time. But as their descendants expand, intermarry, move, and replace or absorb neighbours, their ancestry spreads like ripples in a pond until, many millennia later, millions of us carry faint traces of their DNA. So when you read that a ghost lineage is in millions of people, you shouldn't picture a hidden subspecies living among us. You should picture a long-extinct population that intersected our family tree at a few key moments, and then let basic arithmetic, population booms, and endless shuffling of genomes do the rest. Which brings up the obvious next question. Does any of this actually matter for how our bodies work today? Or is ghost DNA just a historical curiosity hiding in the background of our genomes? It depends what you mean by matter. On one level, every bit of DNA that survived into the present has passed through a brutal filter. It either helped, did no harm, or at least wasn't bad enough to get purged completely. On another level, most of what we've inherited from archaic humans and ghost lineages is boring. Not bad, not good, just background noise. Let's start with the parts that really do something obvious. We already know some introgressed DNA affects traits in living people. Neanderthal and Denisovan segments have been linked to immune responses, metabolism, skin physiology, even how our bodies handle sunlight. Some Neanderthal-derived genes help modern immune cells recognize certain pathogens more quickly. That makes sense. When our ancestors left Africa and met Neanderthals, they also met Neanderthal germs, viruses, bacteria, parasites that African populations hadn't evolved with. Borrowing a few immune genes from Neanderthals was like copying homework from someone who had already taken the class. Denisovan DNA seems to have done something similar at high altitudes. A famous example is a gene region called EPAS1 in Tibetans. It helps regulate how your body responds to low oxygen. The Tibetan version of this gene looks like it came from Denisovans, or a closely related archaic group. If you live your whole life on a plateau 3 miles above sea level, that's not a fun optional upgrade. That's survival. There are hints of archaic influence in how we process fats and carbohydrates, how easily we get inflammation, maybe even in sleep cycles tied to daylight at northern latitudes. Some of these variants may have helped early humans adjust to new diets and new climates as they spread across the planet. So, in principle, ghost DNA could be doing the same thing. If an unknown lineage was adapted to a particular pathogen, or to extreme cold, or to a weird nutrient-poor environment, and our ancestors mixed with them, we might have picked up genetic shortcuts, pre-tested survival hacks built inside their genomes. But here's the catch. For every nice, clear-cut example scientists can point to, there are thousands of archaic fragments that seem to do nothing we can easily measure. When researchers map archaic ancestry across the genome, they see three big categories. First, big deserts, long stretches of our DNA where Neanderthal and Denisovan segments are almost completely missing. That suggests that when archaic DNA landed in those regions, it was often harmful, or at least less good than what was already there, and natural selection pushed it out. These deserts tend to overlap with genes involved in things like brain development, reproduction, and very finely tuned regulatory networks. Second, neutral background. Huge amounts of archaic DNA are just floating around in the genome, neither strongly favoured nor strongly rejected. Over time, recombination chops those fragments into smaller and smaller pieces. They drift up and down in frequency, mostly by chance. They're passengers, not drivers. Third, little islands of enrichment. Spots where archaic-derived variants are more common than you'd expect by luck alone. That usually means selection helped them spread because they conferred some advantage in some environment, at least for a while. When people talk about ghost DNA in millions of people, they're usually talking about that second and third category, mostly neutral fragments, plus a few candidates that might affect traits like immunity or metabolism. And this is where the hype starts to outrun the evidence. It's tempting to say things like this gene from a mysterious human species makes modern people better at X. It sounds dramatic. It makes a great headline. It also skips several steps. To really argue that an unknown lineage shaped a modern trait, you'd need a whole chain of evidence. You'd have to show that a particular segment of DNA in living people really does come from a very divergent source, not just from normal variation within Homo sapiens, or from Neanderthals or Denisovans we've already sequenced. Then you'd need solid data linking that segment to a specific trait, not just a weak statistical blip, but repeated studies showing that people with variant A consistently differ from people with variant B in some measurable way: immune response, oxygen handling, fat storage, whatever. Then you'd want to show that this variant has been pushed around by natural selection. Maybe it's unusually common in one environment, rare in another. Maybe there are telltale patterns around it in the genome that scream recent advantage. Only after all that would it be reasonable to say this ghost lineage fragment probably mattered for adaptation. We're just starting to be able to do this reliably for Neanderthal and Denisovan DNA, where we actually have ancient genomes to compare against. Doing it for a lineage we've never sequenced is much harder. Missing data is part of the problem, but there's a deeper issue too. Traits are almost never controlled by a single bit of archaic DNA. Height, disease risk, brain function, behavior, these things are influenced by thousands of genetic variants, plus environment, diet, stress, culture, random developmental noise. An archaic variant might nudge your immune system to react a little faster to some pathogen. But whether that translates to you get sick less often depends on the pathogens you actually meet, the medicines you have access to, your nutrition, your sleep, your stress levels, and on and on. So when you hear claims like Neanderthal genes make you more anxious, or ghost DNA makes some people better athletes, it's worth being skeptical. Most of those stories are built on early, noisy associations that may not hold up, or that explain a tiny fraction of the trait. And there's one more thing that has to be said, especially when we're talking about genetics and human differences. Human genetic variation does not line up neatly with modern social categories. Populations blur into each other, genes flow across borders. You can have more genetic similarity between two people from different races than between two people from the same one. The clusters geneticists work with are about shared ancestry and geography over long time scales, not about socially defined identities. So even if a ghost lineage did contribute something biologically important, a piece of immunity, a metabolic tweak, that wouldn't make any modern group more primitive or more advanced or more anything in some ranking of human worth. Genes are tools. Different tools can work better in different environments. None of them says anything simple about intelligence, morality, or value. Right now, the safest way to think about ghost DNA is this. Most of it is silent history, background static recording ancient meetings between populations. A small fraction probably did help our ancestors survive in particular places at particular times. An even smaller fraction might still be affecting health and disease risk today in ways we're only beginning to understand. But until we can connect specific ghost-like segments to specific functions with strong evidence, treating them as magical superpowers or as explanations for complex human differences is more science fiction than science. Still, if these unknown lineages really left traces in our genomes, there's a good chance they also left traces in the ground. Which raises the next mystery. If millions of us carry their DNA, why haven't we found their bones? So where do you even start looking for the bones of a species that only shows up in statistics? Not everywhere. Not randomly. Researchers play the odds. They start with understudied regions. Places that, for political, logistical, or just plain historical reasons, haven't had decades of systematic excavation. Central Africa, parts of West Africa, deep interior regions of India and Southeast Asia, high-altitude plateaus that are hard to reach and harder to work in, vast stretches of land where we know prehistoric humans lived because people live there now, because tools turn up on the surface, because the climate would have allowed it, but where actual fossils are rare. Then there are new cave sites. Caves are time capsules. Stable temperatures, protection from rain, slow drip of minerals that can entomb bones in rock. Many of our best Neanderthal and Denisovan remains came from caves. So teams are constantly surveying limestone regions, sinkholes, collapsed caverns, anything that might hide a sealed chamber full of bones and tools. But here's the first big problem. Even if you find a fossil, you still have to recognize what you're looking at. The way paleoanthropologists classify fossils is mostly by morphology, the shape and proportions of bones and teeth. How thick is the brow ridge? How globular is the brain case? How big are the molars? What's the angle of the jaw? Does the pelvis look more like this known specimen or that one? That sounds straightforward until you realize human evolution is not a neat sequence of clearly separated types. It's a messy, overlapping swarm of populations mixing and diverging over hundreds of thousands of years. Morphology reflects two things at once: ancestry and adaptation. If two groups are closely related but living in very different environments, their bodies can diverge rapidly. If two groups are more distantly related but solving similar survival problems, same climate, same diet, same activity patterns, their skeletons can converge and look surprisingly similar. Throw in interbreeding and you get mosaics, skulls with a modern human-like face but archaic-looking brain case, jaws that look primitive attached to otherwise modern skeletons, teeth that don't fit any neat category. So what do you call a fossil like that? For decades, many of them were folded into big, fuzzy species labels. early Homo sapiens, archaic Homo sapiens, Homo heidelbergensis, regional variant. Sometimes the same fragment has been reclassified multiple times as theories shift. Now imagine one of those ambiguous fossils belonged to the very population whose DNA we're calling ghost lineage. It's entirely possible we've already held those bones in our hands. We just didn't know. Misclassification cuts both ways. A genuinely distinct lineage could get lumped into a known species because its bones fall within the broad range of variation we already accept. Or a perfectly ordinary, slightly odd-looking individual could get hyped as a new human type, when genetically they'd fall right in the middle of known diversity. That's why there's so much interest in methods that don't rely just on eyeballing skull shapes. DNA would be ideal. Extract it from the fossil, sequence it, and directly ask, does this genome match our mysterious signal? Is this the ghost? But ancient DNA doesn't survive well in most of the places we care about. Heat, humidity, and time shred it. So researchers have turned to a different biological archive: proteins. Your bones and especially your teeth are loaded with long-lasting proteins. And while DNA is the full instruction manual, proteins are like a heavily abbreviated summary written in a simpler alphabet. You lose detail, but you still keep some information about which genes were there. A field called paleoproteomics has started reading those protein sequences from ancient remains. Dental enamel, the ultra-hard coating on your teeth, turns out to be particularly useful. It's the hardest substance in the human body and can preserve protein fragments for well over 1 million years in the right conditions. By comparing the amino acid sequences in those ancient enamel proteins to living humans, Neanderthals, Denisovans, and other primates, scientists can estimate how closely related a fossil is to each branch. It doesn't give you the whole genome, but it can place a specimen on the family tree when DNA has completely rotted away. It was enamel proteins, for example, that helped show some very old European fossils were closer to Denisovans than to Neanderthals, even though they lived far to the west of the Denisova cave itself. Now extend that logic. If we find teeth from a population that's clearly not Neanderthal, not Denisovan, not modern human, but still distinctly human, and very divergent, that could be a candidate for the ghost lineage behind some of these genetic signals. But we don't even have to find the bones themselves anymore. There's another trick: sediment DNA. When humans or animals live in a cave, they shed DNA constantly: skin flakes, hair, bodily fluids, microscopic fragments of feces and urine. Over time, some of that DNA sticks to mineral particles in the soil. Even if every bone decomposes or is carried away, ghostly traces of genetic material can remain. With careful sampling and ultra-clean lab work, scientists can now extract and sequence DNA directly from cave sediments. That's how Denisovan DNA was identified in layers of Denisova cave that had no visible Denisovan bones. The dirt remembered what the bones could no longer show. This kind of environmental DNA can tell you a hominin was here at this time, and its genome was more similar to X than to Y. In principle, it could even pick up signals from a lineage we've never seen before in the fossil record. You might think, perfect, we just need one good fossil or one clean sediment sample that matches the ghost DNA pattern, and mystery solved. Not quite. The problem is that the ghost signals inferred from modern genomes are statistical composites. They represent an average picture of a population that contributed DNA at some point in the past. But that population itself probably wasn't genetically uniform. It would have had regional variation, subgroups, maybe centuries or millennia of its own internal mixing. If we dig up one individual from one place and one time and sequence them, they might only partially match the ghost signature. Close enough to be related, but not identical. Or they might be from a sister population, a cousin lineage that shared an ancestor with the ghost group, but isn't the exact source of the DNA segments we see today. Even worse, the same ghost, like signal in our genomes, might actually come from more than one archaic population that our methods are currently blurring together. A single fossil can't unmix that. Morphology won't bail us out either. A skull can look weird without being genetically very distant. Or it can look surprisingly ordinary while still carrying deep genetic divergences. There isn't a one-to-one mapping between bone shape and evolutionary distance. So when we finally find a fossil that seems to sit in the right place on the family tree, somewhere between known species, matching the timing and geography implied by the genetic data, it will give us a huge piece of the puzzle. But it may not be a clean, aha, this is the ghost moment. It might be more like, here is one face from a whole cloud of ancient populations our tools are only just beginning to resolve. It narrows the options. It anchors our models. It shows us that these lineages were real, physical beings, not just mathematical phantoms. But it won't, by itself, erase the uncertainty. To really connect ghost DNA to bones in the ground, we'll need all of it working together. Better fossils, more proteomic data, more sediment DNA, and more refined models of how populations moved and mixed. And even then, we may never get a complete set of skeletons for every ancient group our genomes whisper about. Which is why researchers have started to rely more and more on something even stranger than fossils or cave dirt. Traces of DNA floating in the environment itself, recording presences we can't see at all. So how does DNA actually end up in the dirt in the first place? Think about what it means for a group of humans to occupy a cave for a few thousand years. They sleep there. They huddle against the walls. They butcher animals on the floor. They go to the bathroom in the back. Children crawl around. People cough, sneeze, bleed, have sex, give birth, die. Every one of those activities releases microscopic material, skin cells, hair shafts, blood droplets, saliva, fecal particles, urine crystallizing into salts. Each tiny fragment carries DNA. Most of it decomposes fast. Bacteria chew it up, water washes it away. But in dry, cool, mineral-rich environments, like deep cave sediments, some DNA fragments stick to clay particles or get trapped inside tiny pores in minerals, or adsorb onto bits of charcoal and bone dust. Layer after layer of sediment builds up. Dust blown in from outside, ash from fires, collapsed ceiling fragments, trampled soil. Mixed into those layers is this invisible snow of genetic debris from every creature that passed through. Humans, wolves, cave bears, bats, rodents, plants, even microbes. That's the archive. Extracting it is like trying to read a book that's been shredded, soaked, and then stirred into mud. Researchers take small, carefully chosen samples from specific layers of sediment, often just a few grams. In ultra-clean labs, they dissolve the dirt in chemical solutions that release whatever DNA is stuck to the minerals. Then they use molecular hooks designed to latch onto mammalian DNA, or more specifically hominin DNA, pulling those fragments out of the soup. What they get is not long, pristine sequences, it's tatters, 30, 40, maybe 60 base pairs long, heavily damaged, full of characteristic chemical scars from thousands of years of decay. Computer algorithms then try to match those tiny snippets to known genomes, modern humans, Neanderthals, Denisovans, other animals to figure out who was there. When it works, the results can be astonishing. At Denisova Cave, for example, some layers contained no visible hominin bones at all. No skulls, no jaws, just animal remains and stone tools. But the sediment DNA said otherwise. Certain layers lit up with Neanderthal-like sequences. Others carried a clearly Denisovan-like signal. In a few, both lineages were present in the same time slice, implying overlapping or alternating occupations. In other caves across Eurasia, sediment DNA has revealed Neanderthals in layers where no Neanderthal bones were ever found, or confirmed that modern humans had arrived even though their skeletal remains were missing or fragmentary. In some cases, it's even hinted at sex ratios, more female than male. Neanderthal DNA in a layer, suggesting that particular group was female biased, maybe because of how they moved between bands. Suddenly, caves that looked empty of human fossils aren't empty anymore. The dirt remembers. The big strength of sediment DNA is exactly that breadth. Bones are a lottery. You get whoever happened to die in that particular spot under the right conditions. Sediment captures everybody who lived there, even if they left no skeletons behind. It can reveal repeated occupations across thousands of years, track which species came and went, and show how human presence lined up with climate swings, animal communities, and tool traditions. But the trade-off is brutal detail. DNA in sediments is insanely fragmented. You rarely get enough from a single layer to reconstruct a full genome. Often you're working with low coverage, just enough to say this looks more like Neanderthal than modern human, or this is clearly hominin, but doesn't match any reference perfectly. Contamination is a constant threat. Modern human DNA is everywhere, from archaeologists, tourists, local animals, even drifting dust. Labs fight this with clean suits, filtered air, chemical treatments that preferentially destroy recent DNA, and statistical methods that look for the specific damage patterns that mark genuinely ancient sequences. Still, you have to be careful. A single misplaced fingerprint can masquerade as ancient human presence if you're not vigilant. So sediment DNA is powerful but blunt. It tells you someone was there, and roughly who they were related to. It doesn't hand you a clear named species with a face. This is where environmental context becomes crucial. Sediment samples aren't taken in a vacuum. They're anchored to layers dated by radiocarbon, uranium series, or other geological methods. Those layers are packed with clues. Animal bones that tell you the climate, cold steppe versus warm woodland, pollen grains that reconstruct vegetation, charcoal from fires, specific tool types. Put it all together and you get a scene. A cave layer dated to 200,000 years ago in a glacial phase, with cold-adapted fauna and a particular style of stone tools that elsewhere are associated with Neanderthals. If the sediment DNA in that layer shows an unknown hominin signal that's closer to Neanderthals than to modern humans, your list of candidate populations tightens. A tropical cave in Southeast Asia, 50,000 years ago, with remains of island fauna and a tool tradition that doesn't quite match known modern human industries, turns up hominin DNA that's clearly not Neanderthal and not Denisovan. Now you're looking at a different set of possibilities. Maybe a regional archaic population, maybe something that contributed to the ghost signals we infer in certain modern groups. The environment acts like a filter. We know roughly which hominins were where and when, based on the combined fossil and archaeological record. When sediment DNA points to something hominin, in a layer that falls outside those expectations, wrong place, wrong time, wrong genetic affinity, that's when ears perk up. That's when people start whispering about unknown lineages. In that sense, sediment DNA could be the bridge we've been missing between abstract genetic models and the stubborn absence of fossils. Geneticists working from modern genomes can say, there was a deeply divergent population that mixed with the ancestors of this group, probably in this broad region, roughly at this time. Paleoanthropologists working from caves and sediments can say in this cave, at this time, there was a hominin whose DNA doesn't quite match Neanderthals, Denisovans, or us, living in this specific ecological setting, using these tools, hunting these animals. Neither line of evidence on its own is decisive. Ancient DNA from bones is still rare. Skeletons are patchy and ambiguous. Sediment sequences are short and noisy. But when all three lines start to converge, modern ghost signals odd sediment DNA, and a particular environmental and archaeological context, you inch from speculation toward a coherent story. You can imagine a near future where a ghost lineage is inferred from African genomes. A cave in Central Africa, long known for its stone tools but empty of hominin fossils, yields sediment DNA that doesn't cleanly match any known reference. The layer is dated to the right window. The ecology fits what models predict. Proteins from a few stray teeth in the same layer place their owner on the tree in just the right spot, off the main Homo sapiens line, but clearly close enough to have interbred. No complete skull, no famous fossil face. And yet, piece by piece, the dirt, the tools, the proteins and the ghost signals begin to describe a real population. Not a mathematical phantom, not a mythic lost race, just another group of humans who lived, died, and quietly left their DNA in the dust. Which brings us back to those ghost signals themselves, because even with sediment DNA, we still have to know what we're looking for. And that means understanding how these mysterious introgressed fragments actually show up in our own genomes, and what, if anything, they're doing there. When people hear ghost species, the imagination goes straight to science fiction, a secret population of almost humans watching us from the shadows, a hidden branch of the family that somehow survived in a jungle valley or inside a mountain, breeding in isolation for hundreds of thousands of years, and then vanishing, leaving only a few drops of DNA in our veins. It's a compelling image. It's also almost certainly wrong. The genetic ghosts we're talking about are not a single, coherent, hidden tribe. They're not a community that somehow escaped every fossil site, every sediment layer, every archaeological hint. What the data actually point to is much messier, and in some ways, much more interesting. Those strange DNA fragments we see in modern genomes probably came from populations that looked from the inside completely ordinary. Small bands of humans or near humans moving across landscapes, forming alliances, fighting, trading, having kids. Some of those kids inherited stretches of DNA that by chance and by usefulness stuck around in certain descendant groups. Over hundreds of thousands of years, most lineages go extinct. Most genomes vanish without a trace. Ghost just means this particular lineage left DNA in us, but we haven't yet matched it to a clear fossil series or a fully sequenced ancient genome. That's all. When headlines say a human species that doesn't exist, what they really mean is a population we haven't yet identified by bones and a name tag. Their genetic signature is real enough. What's missing is a concrete fossil in a museum drawer labelled with a neat Latin binomial. Even the word species here is slippery. In modern biology, species are often defined by reproductive isolation. You can't easily have fertile offspring across the boundary. But ancient humans didn't follow our textbook categories. Neanderthals and Denisovans interbred. Modern humans interbred with both. Some of these ghost signals come from groups that were clearly able to mix with our ancestors. So are they different species, or just very distinct populations on the same spectrum? Geneticists argue about this. Paleoanthropologists argue about this. The only thing everyone agrees on is that the simple picture book family tree with clean branches labelled Homo sapiens, Neanderthal, Denisovan, mystery species X doesn't really capture what was going on. And this is where pop culture tends to go off the rails. The narrative usually jumps straight from "we see unexplained DNA" to "scientists have discovered a completely new human species that overturns everything we thought we knew." But in the actual papers, the language is far more cautious. You'll see phrases like "deeply diverged archaic source," or unsampled hominin population, or introgression from a lineage related to. You'll see confidence intervals. Maybe this mixing happened 50–70,000 years ago. Maybe the split from other humans was 500–800,000 years before that. Maybe the contributing population was widespread, or maybe it was localized. Everything is wrapped in ranges and maybes, because that's how this works. Genetic signals can often tell you that something happened. Some group mixed with the ancestors of this population, leaving behind DNA that doesn't match any known reference. Pinning down exactly who, where, and when is harder. And sometimes alternative explanations exist. Ancient population structure within Africa, small, long, isolated groups of Homo sapiens, or even quirks of the models used to detect introgression. To their credit, many researchers spell out these uncertainties in painful detail. But by the time the story becomes a headline, most of that nuance has been stripped away. So how do you think better about ghost species claims? First, translate the language. If you see unknown human species in an article, silently replace it with a population we haven't sampled directly. That's almost always closer to what the data actually show. Doesn't exist should become not yet identified in the fossil or ancient DNA record. Second, look for the caveats. Good scientific communication will tell you things like, how strong is the signal? Is it a tiny fraction of the genome or a big chunk? Is it seen in multiple independent datasets, or just one research group's analysis? Are there competing explanations the authors take seriously? If the story you're reading doesn't mention uncertainty, rival hypotheses, or error bars, it's probably oversimplified. Third, pay attention to what would actually change minds. Scientists don't cling to ghost explanations out of romance. They use them because, given the current evidence, a separate, unsampled population fits better than the alternatives. But that could change with new samples. A single, well-preserved bone with extractable DNA from the right time and place can suddenly turn a ghost into a named lineage. That's what happened with Denisovans. First a weird genetic signal, then a finger bone, then teeth, then sediment DNA, all slowly knitting together a picture. Better models, methods for detecting ancient admixture are constantly being refined. If a new approach explains the same patterns without invoking an extra population, say, by modeling more realistic structures within early Homo sapiens, people will test that hard. Replication. When independent groups, using different datasets and methods, keep finding the same ghost-like signal, confidence grows. When results wobble or disappear under slightly different assumptions, enthusiasm cools. The real story of ghost lineages isn't a secret people hiding from us. It's a moving boundary between what we've directly sampled and what we can only infer. And you can use that to filter future headlines. When the next article pops up saying, "Scientists discover mystery human that interbred with us," ask a few quick questions. Did anyone actually sequence DNA from new bones, or is this inferred from modern genomes alone? Are they talking about one study or a pattern that multiple groups have reported? Do the scientists quoted use careful, hedged language, or are the boldest claims coming from the journalist and the title? Is there any mention of alternative explanations, or is it framed as a single dramatic revelation? If it's all certainty and no context, you're not getting the real picture, because the real picture is already strange enough. We know that our lineage went through a brutal bottleneck. We know it then split into multiple branches. Some we've named, some we haven't. We know those branches met and mixed over and over, trading genes and ideas. We know that fragments of those encounters are still inside us, influencing everything from altitude tolerance to immune responses. Some of those fragments come from groups we can point to in caves and museum drawers. Others, for now, are just statistical whispers, Looking at that landscape, the right reaction isn't secret species hiding in the shadows. It's this family tree is a lot more tangled than we thought. And if ghost lineages are the blurry parts of that tangle, the next step is obvious. Stop thinking of our past as a simple branching diagram and start thinking of it as something else entirely. So let's do that. Let's retire the comforting family tree for a minute and swap it for something closer to reality. Instead of a clean trunk that splits into tidy branches, imagine a river delta. One channel divides, then those channels reconnect, split again, braid together, disappear, underground, re-emerge somewhere else. Water is constantly finding new paths, sometimes merging back with streams it separated from thousands of twists ago. That's what human evolution looks like once you take gene flow seriously. The old picture, one ancestral species giving rise to another in a neat, one-way branching pattern, assumes that once two lineages split, that's basically it. They go their separate ways, maybe competing, but not really mixing again. The genomic evidence is telling us the opposite. Neanderthals and Denisovans split from our ancestors, adapted to radically different environments, accumulated hundreds of thousands of years of differences, yet they were still close enough that when their descendants bumped into ours, they had kids. Some of those kids survived, had more kids, and now their DNA shows up in our genomes, and ours in theirs. And the ghost lineages, they push this even further. They tell us those braided streams weren't just between the big three we've given names to. There were side channels, unsampled groups, dropping genes into the main flow and then disappearing. In evolutionary terms, this is called reticulate evolution. Not a simple branching tree, but a network with loops and crosslinks. That has huge implications for how we think about species. Because if you flip open a textbook, species is often presented as this bright, sharp line. On one side, you can interbreed and produce fertile offspring. On the other, you can't. Easy, clean, but the Pleistocene did not care about our definitions. What we see instead is a spectrum. Populations could be quite distinct, different skull shapes, different bodies, different cultures, and still partially compatible. Interbreeding might have been rare, maybe only under certain ecological or social conditions, but it wasn't impossible. So what do we call those groups? If you insist that species must never successfully interbreed, then Neanderthals, Denisovans, and early Homo sapiens arguably weren't separate species at all. They were long separated, strongly structured populations of one very flexible species complex. If you define species more by long-term evolutionary independence, distinct histories, distinct adaptations, then they qualify as separate species even though their boundaries were leaky. Either way, the human past starts looking less like a set of clear boxes and more like a set of overlapping clouds. Edges blur, boundaries leak, genes cross lines we thought were solid. And once you accept that, the story of modern human origins shifts too. For decades, debates were framed as either-or, a strict out-of-Africa replacement, where Homo sapiens evolved in one small region and then spread out, overwhelmingly replacing other humans, or a multi-regional model where modern humans emerged more gradually across the old world from interlinked archaic populations. The genomic data we have now point to something in between and more dynamic. Yes, there is strong evidence that a major wave of Homo sapiens expansion came from Africa relatively recently in evolutionary terms, around 60,000–70,000 years ago. Yes, most of the genetic ancestry outside Africa traces back to that wave. But that wave didn't expand into a vacuum. It moved into landscapes already occupied by Neanderthals, Denisovans, and probably other regionally distinct groups. It mixed with them to varying degrees. It picked up helpful genes and maybe lost some of its own. It was shaped in part by what was already there. Inside Africa, things were likely even more complex. Instead of one small cradle where modern humans suddenly appear, many researchers now picture a patchwork of semi-isolated populations across the continent, each region experiencing its own environmental pressures, its own small experiments in skull shape, behavior, body proportions. These groups split, reconnected, exchanged genes, and drifted apart again over hundreds of thousands of years. In that view, modern humans weren't born in a single birthplace at a single time. They coalesced out of a network of different African populations, some of which may now look ghost-like to us in the DNA, contributing pieces to what eventually becomes the global Homo sapiens pattern. Our origins, then, are both regional and shared. Regional because different places contributed different ingredients. Shared because those regions were never completely cut off. They kept leaking genes into one another, and eventually into everyone alive today. And we're still just sketching the outlines. If ghost lineages are real and multiple independent studies keep pointing that way, then we've only mapped a fraction of the web. Obvious questions follow. How many of these unsampled lineages were there? Were they long-lived, widespread populations, or small, short-lived groups that just happened to leave descendants in one corner of the world? Did they mostly live in Africa, where the fossil and ancient DNA records are sparse? Or were there Eurasian groups we simply haven't recognized yet? When exactly did they branch off from other humans, and when did they mix back in? Right now, many of those questions have only wide ranges for answers. Somewhere between X and Y 100,000 years ago. Possibly in this region, but the signal could be consistent with that other region too. And this is where the next big step might surprise you. You might imagine that to uncover another ghost lineage, we need a spectacular new fossil find from some untouched cave. That could certainly happen. One well-preserved jaw or skull or toe with good DNA could rewrite whole chapters. But there's another, quieter frontier, reanalyzing data we already have. Genomes that were sequenced 10 years ago, when methods were cruder, can be re-examined with new statistical tools. Ancient DNA datasets that were initially scanned only for Neanderthal or Denisovan ancestry can be probed for more subtle, off-tree patterns. Fossils sitting in museum drawers can be redated, remeasured, or sampled again with improved techniques. Sometimes the ghost isn't hiding in a remote jungle. It's hiding in a spreadsheet on someone's hard drive, or in a bone fragment that was labelled and filed away before we knew what questions to ask. As models of population structure improve, as reference genomes from living and ancient people grow, some signals that currently look like mystery species may resolve into more mundane explanations, subtle patterns within Homo sapiens, for example. Others, however, may sharpen, becoming harder to explain without invoking genuinely distinct, unsampled lineages. Either way, the direction of travel is clear. The more clearly we see, the less human evolution looks like a straight line or a single branching path, and the more it looks like a woven mesh of populations, splitting, reconnecting, and sometimes leaving just enough of themselves behind that their existence can still be detected in us. Which leaves one final puzzle. If our past is really that tangled, how do you actually prove that a particular ghost lineage was real and not just a clever statistical mirage? To answer that, we have to zoom out for a second. Because this problem, trying to reconstruct something real from faint, conflicting traces, isn't unique to ghost DNA. It shows up all over science and history. We're basically doing forensic work on reality itself, taking scraps of evidence, patterns that don't quite fit, and asking, is this noise, or is this the outline of something we haven't named yet? That's not just a human evolution story. It's the same pattern behind a lot of the most intriguing unsolved puzzles we've ever run into. If you like this kind of thing, where the data are real but the explanations are still open, then I put together something you might want to dive into after this video. On my website, there's a book that walks through 12 of the most fascinating unexplained mysteries we have good evidence for. Not conspiracy stuff, not because aliens, but real stubborn anomalies that serious researchers still argue about. Some of them are cosmic. Signals in the sky that don't behave the way we expect. Strange regularities in the universe that hint we might be missing a piece of the underlying rules. Some are geological and environmental. Sudden climate lurches in Earth's past, mass die-offs that don't line up cleanly with any of the usual suspects, traces of catastrophic events that might have reshaped whole civilizations. Others are closer to what we're talking about here. Odd genetic patterns, archaeological outliers, and cultural puzzles where different lines of evidence point to something, but not quite the same. Something. The common thread is exactly what we've been circling around in this video. We can see the fingerprints, but the hand that made them is still in the dark. Each chapter takes the same approach we're using with ghost lineages, lay out what's actually known, where the data come from, what the mainstream interpretations are, and then where those interpretations start to wobble. No hype, no certainty where there isn't any, just the best current thinking and the open questions that still keep people working on them. If you're the kind of person who hears ghost population and immediately wants to know what else is lurking in the datasets we thought we understood, that book is basically 12 deep dives into that feeling. Right now it's discounted for this week. If you want to grab it, the link is in the description and it'll take you straight to the page on the website with all the details, formats, and a preview of the chapters. You don't have to hit pause. You can bookmark it for later, finish the video, and then go lose an evening thinking about time-slipped climate events, maybe two regular cosmic coincidences, and archaeological sites that refuse to fit neatly into any timeline. Because here's the connection back to our invisible hominin neighbours. Each of those 12 mysteries is another example of how reality leaves us partial records. Tree rings with missing years, ice cores with weird spikes, genomes with segments that don't match anything in our reference panels, maps with blank patches labelled, "Here be something," but we're not sure what yet. In every case, the question is the same. Are we looking at a new kind of thing, or an old kind of thing, behaving in a way we just haven't seen enough to recognise? That's exactly where ghost lineages sit right now. We have these stretches of DNA that don't fit cleanly into any known branch. They're too divergent to be regular variation within Homo sapiens, but they don't line up nicely with Neanderthal or Denisovan either. They look like imports from a population we haven't sampled. So what do we do with that? Do we start drawing new branches on the family web and giving them provisional names? Or do we treat them as artifacts of our models until we get something harder, like a fossil with usable DNA? And how close are we, realistically, to moving this unknown group from the ghost column into the "we can describe their history, their range, maybe even their faces" column? Because we're not as far away from that as it might seem. Every year, sequencing methods get more sensitive. Labs pull usable DNA out of older, more degraded bones. New statistical techniques peel apart signals that used to blur together. Sediment DNA can now tell us that some kind of human was in a cave, even if not a single recognizable bone remains. At the same time, archaeologists are turning up fossils that don't fit neatly into our named categories. Skulls that look partly modern, partly archaic, jaws with weird mixes of traits, teeth that might reflect local experiments in being human that we haven't properly slotted into the web yet. The big looming question is whether any of those odd fossils, any of those unsatisfying labels in museum drawers, actually belong to the same populations whose genetic echoes we're seeing in living people. If they do, then the ghost stops being a pure abstraction. It becomes a population we can point to in space and time. So that's where we're headed next. What lines of evidence do we have right now that let us narrow down when this unknown group branched off, where they might have lived, and how often they met and mixed with our ancestors. How close are we to being able to say with a straight face, this is probably the source population behind that mysterious DNA? And just as important, what would it take to go from probably to we're actually confident about this? So let's pull all of this together and be brutally clear about where we actually stand. On the "this really looks like a ghost lineage" side of the ledger, we've got several independent lines of evidence, all pointing in the same direction. First, the divergent signal. When geneticists look at certain chunks of DNA in people today, especially in some African populations, they find sequences that are way more different from typical Homo sapiens variation than you'd expect if everyone's ancestry came from a single continuous human population. These segments look like they split off from the human line hundreds of thousands of years earlier than the rest of our genome suggests. Second, the too old for comfort coalescence times. You can use patterns of mutations to estimate when two copies of a gene last shared a common ancestor. For most of our genome, those dates cluster in ranges that make sense if our main lineage went through bottlenecks and expansions we already know about. But some pieces look like they separated so long ago that they'd require a population hanging off the side of the family tree, isolated for a very long time, then mixing back in. Third, the shape of the DNA blocks themselves. These suspicious segments aren't sprinkled randomly. They tend to come in longer, relatively intact stretches in some people, and are nearly absent in others, which is what you expect from a pulse of gene flow from a distinct population, not just ancient standing variation drifting around within one big group. And fourth, the fact that similar methods used on Neanderthal and Denisovan DNA successfully predicted their existence before we had any clue what they were. The exact same class of models that once said, there's archaic DNA in modern Eurasians long before anyone sequenced an actual Neanderthal genome, are now saying, there's something else here we haven't sampled yet. That's a strong pattern. Different methods, different teams, different datasets, all converging on, there was at least one population that doesn't match any of our named species, and their DNA is still in us. But on the other side of the ledger, there are also valid reasons to be cautious. One big one is model dependence. Every time you reconstruct history from genomes, you have to make assumptions about mutation rates, about how populations split, about how much they mixed, about how many hidden groups you allow the model to have. Change those assumptions and some ghost signals weaken or disappear. A few unknown archaic results vanish if you let Africa be more structured, more patchy, with semi-isolated groups without invoking a separate species. Another issue is reference bias. Our maps of Neanderthal and Denisovan variation are still based on a tiny number of fossils from specific caves. We don't have a Neanderthal genome from every region they ever lived in, or Denisovan DNA from all across Asia. It's possible that some of the unknown DNA is just Neanderthal-like or Denisovan-like variation from populations we haven't sampled yet. In other words, not a whole new species, just missing pages from lineages we already know about. There's also the problem of convergent patterns. Certain demographic histories, like a long period of deep structure within Africa, where different groups were partly separated but never fully isolated, can generate DNA patterns that look eerily similar to archaic introgression. In some simulations, if you make early Homo sapiens in Africa very patchy and long-lived, you can get ghosty signals without needing a separate, long, isolated hominin. So we're left in this uncomfortable middle. The simplest story, the one that fits a lot of the data, is that there really was at least one archaic population we haven't sampled directly, probably more than one that contributed genes to the ancestors of some modern humans. Not just Neanderthal, not just Denisovan. Something else, or several something elses, blurrier and more local. But there are other plausible scenarios. One scenario is unknown population, not quite a separate species. Imagine early Homo sapiens in Africa split into very divergent regional groups for hundreds of thousands of years. Some of those groups became so genetically distinct that if we met them today, we might be tempted to label them a different species. Others stayed more connected. This deep structure, combined with periodic mixing, could give you DNA that looks very old and very weird without requiring a fully separate lineage like Neanderthals. Another scenario is multiple sources smeared together. Instead of one ghost, imagine several. A little gene flow from a very archaic population in one region, plus some from a late surviving cousin in another, plus deep structure within Homo sapiens itself. Over time, recombination scrambles these signals, and our models, which like neat, simple stories, compress that mess into one ghost population. In reality, the web might be much more tangled. A third scenario is our definitions need work. Maybe what we're really seeing is that the line between archaic and modern is much fuzzier than the labels on museum walls suggest. Populations split, diverged, and merged again so often that trying to count how many species there were might be the wrong question. The DNA is telling us there were just a lot of semi-isolated experiments in being human, some of which left more traces than others. So, what would it take to move from "these are good stories" to "we're genuinely confident about what happened"? 3 kinds of data would change everything. First, more ancient genomes from the right times and places. We have a handful of African genomes older than 15,000 years. That's basically nothing. If we start pulling high-quality DNA from skulls and bones in Africa that are 50,000, 100,000, 200,000 years old, we can directly see whether any of them carry the same mysterious segments we see in living people. If we ever sequence a fossil that looks morphologically odd and its DNA turns out to be a near-perfect match for the ghost signal, the debate ends. At that point, the unknown population isn't unknown anymore. Second, much denser sampling of living humans, especially across Africa. A lot of the most interesting signals are in groups that are underrepresented in current genetic datasets. As more communities choose to participate in research under fair, ethical agreements, the resolution of our picture increases. We might find that what looks like one ghost population is actually several region-specific ones. Or we might see gradients that make deep structure a better fit than a distinct archaic group. Either way, more genomes means sharper tests. Third, more environmental and molecular traces. Sediment, DNA, and proteomics. Sediment DNA lets us say, some kind of human was here, even if we never recover a single recognizable bone. Imagine matching the age of a sediment DNA sample to the estimated split time of the ghost lineage. If cave sediments in a particular region show persistent, strange hominin DNA over hundreds of thousands of years, that's strong circumstantial evidence for a long-lived local population we haven't named yet. Proteomics, recovering proteins and their sequences from old bones and teeth, pushes our reach even further back in time than DNA alone allows. Proteins are more durable, so in fossils where DNA is gone, we might still reconstruct enough of the amino acid sequence to place that individual on the human family tree. If an odd-looking fossil yields protein sequences that branch off at exactly the depth our ghost DNA models predict, you suddenly have a bridge from abstract statistics to a face, a skeleton, a place on the map. Put all of that together, and the next Denisovan moment starts to feel less like a distant possibility and more like a when, not an if. Denisovans went from 0 to fully established in about a decade, first as weird DNA in a pinky bone, then as a named group, then as a key player in the ancestry of millions of people across Asia and Oceania. The same could happen again. One good genome from the right fossil. One cave with unusually well-preserved sediments. One protein sequence that refuses to sit neatly on any known branch. And suddenly, the anonymous archaic component in your genome report has a face, a home range, maybe even a known tool tradition. Until that happens, we're going to stay in this productive tension. Solid hints, competing models, and a story that's still being written. If you want to keep following that story as it unfolds, and you like going deep into these places where the evidence is real, but the answers are still open, remember that book I mentioned earlier. It digs into 12 of these prehistoric and cosmic puzzles in exactly this way. What we know, what we don't, and how we might actually solve them. The link is in the description. It'll take you straight to my website with all the details and a preview of the chapters. And if you found this exploration of ghost DNA and unknown human lineages interesting, this is the part where I ask for the usual help that actually makes a difference. Hit like if this made you think about our origins in a new way. Subscribe if you want more deep dive videos on human evolution, ancient mysteries, and the strange edges of what we think we know. And drop a comment telling me where you're listening from. I love seeing how many corners of the world are carrying these tiny fragments of vanished humans in their DNA. Somewhere out there, in a drawer, a cave wall, or a dusty core of sediment, the next clue is already waiting.